Good morning, everyone. Thanks for joining in. We'll start at 10 o'clock. All right. Good morning, everyone. Thanks for joining in. I just wanted to check before we get started the presentation. I hope all of you have received two emails from me. One was with the Zoom link. The other one was with your cloud login credentials. If you haven't received a second email with your cloud login credentials, please let me know in the chat box. All of you should have received it, but if anyone hasn't received it, please let me know. Thank you. We'll start the session at 10 o'clock. Yeah, thank you. Yeah, just let me know if you haven't received it. You don't have to tell me if you received it or not. Yeah, all the login issues we'll address later. But for now, like if you got the email, that's sufficient. Nothing else to do right now. We'll help you with the login issues later, but the schedule is that we'll first have a presentation from 10 to 10:45, and after the presentation over, then we'll go for the hands-on session. So we'll show you how to use the software. So if you didn't receive the email, let me know. If you have any issues with logging in, don't worry about it. We'll help you later with that. The first presentation. Once that is over, then we'll help you with your login issues. Just be patient. All right, so we'll just wait for 1 or 2 minutes more um, to see if anyone will join slightly late, and then we'll start with the presentation. If you have any login issues that you're facing right now, we'll address them after the presentation is over. All right, let's get started. So I know that many of you are new to this area. So what we'll do is we'll first have a very basic presentation for today that will cover some basic concepts of drug discovery. And we'll also be covering on docking specifically in today's presentation. Then we'll have the hands-on session for various topics. We will guide you through how to use the software, um, get an understanding of the software. Then we'll also show you how to use the software for docking calculations for today. And tomorrow we'll have a much more thorough presentation on all the tools that the platform provides. There are plenty of tools in Studinger. Tomorrow we're going to cover in the presentation many of those tools. And then we'll have a hands-on session for molecular dynamic simulations and machine learning models, predictions, and building. This workshop will be I'll be presenting today and tomorrow for the morning presentation, and then I'll also have my colleagues, Dr. Vinod Devaraji and Dr. Abhijit Kayal, who will help us with the hands-on sessions also along with me. So in this presentation, I would be addressing briefly on the computational tools for drug design. Why do you have to use them? And how you can perform the most basic drug design calculation, just talking calculation. We're going to look into that as well. Just a minute. Yeah. If you have any questions or anything like this, please put the question in the chat box and I'll take the questions as I go along the presentation. If you're having issues with logging in, I'll take care of those issues later. Okay, after the business is over. Yeah. All right. I hope all of you can see my screen. Can you confirm with the slide presentation? Yeah, thank you. So yeah, so in the computational tools for drug design, we're going to be focusing mainly on small molecule drug discovery for this workshop. But be aware that in our software platform, we have tools for designing even protein-based therapeutics. So in the case of small molecule drug discovery, we have a known protein target, and we want to see which molecules, small molecules can bind to the protein target to inhibit the protein. In the case of biologics, our goal is not to see how a small molecule binds to it. Our goal is to optimize the protein itself for various different reasons, endpoints. We won't be worrying about that in this workshop. We'll focus mostly on the small drug discovery. We also have a whole suite of material science where you can work on polymers, nanoparticles, nanotubes, organometallic complexes, very different type of chemistry, including batteries. All of that stuff can be done in the material science suite. Our focus of the workshop will be on this one, small drug discovery. So if you look at the life science module specifically, like what operates on either protein or protein-based therapeutics, you have two different approaches. One is small molecule therapeutics, and you have the biologics and protein-based therapeutics. In either of these cases, you have to these different stages in your process. In the case of small molecule therapeutics, you have to identify the target that you want to inhibit. So this can be a protein or a DNA or RNA molecule, and that's what you want to inhibit in the case of small molecule. And so what you want to do is you want to understand for a given disease, which target to inhibit. So what people often do to figure out which target to inhibit is that they take a normal cell and they take a cell under disease conditions. They try to see which proteins are expressed abnormally in the case of a disease cell, and they compared this gene expression profiles between the two cells to figure out, like, this is the protein that I want to target. And maybe if I kill that particular protein, I can probably prevent the disease that is happening for me right now. So this stage is like most experimental. But once you identified your target protein, they have to know if you have a structure for the protein or not. So in the PDB, there are plenty of structures available. In the protein data bank, there are around 235,000 structures available for different proteins, different RNA molecules, also different DNA molecules also. But there are lots of proteins for which there's no structure available. So sometimes you may have to predict the protein structure, and you may have to refine the protein structure if you don't have it from the PDB. Once you have the protein structure available for you, then you have to identify hit molecules. So hit molecules are those that have a decent potency to bind with your protein. So by decent, I mean like they should bind with a 10 micromolar or better binding potency than that. It can be 10 micromolar to hundreds of nanomolar. Once you identify a hit molecule, then you have to take the hit molecule to lead molecule, and the lead molecule will be binding in the order of, it's a very tight binder. So typically it will binding in nanomolar or picomolar range. And you have to make sure the lead molecule is optimized to prevent issues with adsorption, distribution, metabolism, excretion, and toxicity. And once you have a drug molecule, then you have to design a formulation for the drug molecule. For example, if you go to a pharmacy, you will see that some medicines are in a tablet form, some are in a capsule form, some are, and you have to take a injection for some of them, and some of them you can take it as a syrup. So some of them are powder. So each of these medicines will require different kind of formulation for it. So you have to be able to figure out what is the right formulation for a given molecule. So computational tools can help you in each of these stages: target validation, potential prediction and refinement, hit discovery, hit to lead optimization, and drug formation also. We're going to spend some more time on each of these aspects later. At the same time, if you want to work on protein-based therapeutics like vaccines or antibody design and things like that, we also have tools for predicting your protein structures, antibody structures, and sometimes the starting point for antibody or your protein-based therapeutics can be from a different species like mouse or some other species or yeast. So you'll have to convert that antibody from that other species to a human antibody. And that means that you have to do some mutations on your antibody to make sure your body doesn't have an allergic response to a foreign looking antibody. Just hold on. Yeah. And once you humanize the antibody, you may also want to improve the properties of the antibody. So you have to do some mutations for the proteins. So you have tools for doing protein mutations also. And we also have tools for figuring out if your antibodies or your protein-based therapeutics have some liability issues, similar to the case of lead optimization for small molecules. And eventually you have to formulate your drug molecule or antibody or your protein-based things. Most of the times, biological-based therapeutics, these are solutions. So if you think about your vaccines or if you think about your antibody or any of these protein-based therapeutics, most of the time they're given us through an injection. Our focus for the workshop will be on small molecule therapeutics, but if you're interested in these things, you can reach out to us later by email and we can try to maybe help you out with those queries. So for small molecule therapeutics, we have these 5 different stages: target validation, protein structure prediction refinement, hit discovery, hit to lead and position, and drug formulation. So I'll focus more on these two stages here: hit discovery, hit to lead into this presentation. Tomorrow I'll also be addressing some of the tools for protein structure prediction and drug formulation. But for today, we'll only focus on these aspects here. So if you want to find, like, once you identify your protein target, if you want to find a good drug molecule, traditionally people used to do this, but that was like 10, 15 years ago. And you had three stages of hit discovery, hit to lead optimization. In the case of hit discovery, you try to look for molecules that have a decent potential to interact with a protein in micromolar or triple digit nanomolar range. Then you want to find the lead molecule which has a single digit nanomolar or picomolar range for binding. Then you want to make sure your molecule is optimized to prevent any issues with adsorption, distribution, MET, other things, ADMET. So people used to come with these drug molecules by manual molecule design in the traditional approaches, and there was a limited success for these methods. So what they used to do was they used to take like 5,000 molecules for a given project, roughly the synthesis of 5,000 molecules, and test them over a period of 4 to 6 years. Now you have to keep in mind that every project has a particular timeline, and so these molecules that they used to synthesize were based on intuition of the medicinal chemist. And with that intuition, they're able to identify decent molecules to bind to the protein. And what used to happen in the traditional approaches was that even if they had a molecule with some property not working, they should still propagate that molecule to the next stage because they have to meet their project deadlines within a certain amount of time. So you used to have like maybe some molecule which has like solubility issues that will propagate to the next stage, hoping that you'll eventually resolve that issue later on. But what used to happen was that even if you have one small issue with any of the molecules, that whole drug development candidate will not be cannot be taken forward for clinical trials. So it used to happen that one out of three projects will succeed, and two out of every three projects used to fail. We had a very high chance of failure in these approaches. Nowadays, what people do is they use these computational tools where the idea is you still want to go through the three different stages, but instead of testing like a few thousand molecules, you want to test billions of molecules in various computational assays. And you still go through the stage of hit discovery, hit to lead optimization. But because you start with a much bigger pool of molecules, if any molecule is not having the right property profile, at the stage of discovery, you can discard those molecules and take only the most promising ones to the next stage, and then do your calculations, and then again take the most promising ones to the next stage. So since you start with a much bigger pool of molecules compared to traditional approaches, you have a much higher chance of finding a drug molecule with optimal property profile. Eventually, you still have to go and test these molecules. You have to synthesize and test them. And currently, people test around maybe a few hundred molecules, not more than that, in the wet lab. And the entire process of hit discovery, hit to lead optimization, including preclinical trials, takes 2 to 3 years currently. So things have improved significantly. And the reason why we could do this is because these computational systems are very, very fast and they're very cheap compared to experimental methods. And that's will allow you to screen large number of molecules. And because you have much wider chemistry, even if something fails, you can always figure out some other molecule that might work. So these computational tools have become more and more popular in the last 10, 15 years, and we'll see why that is the case in today and tomorrow's sessions. But the first question I want to ask all of you is, why do you want to use these computational tools? Any ideas? Just ping the chat if you have any ideas as to why you want to use these tools. Why are you here for the workshop? Why do you want to use these tools? Why do you want to learn these tools? Time saving, yes, they save a lot of time because they are faster. Any other reasons? Yeah, if you can save time, you also reduce cost because one is if you can reduce the number of molecules you test experimentally, that will reduce the cost. And secondly, it also will save the manpower that is reduced if you save the time. Yeah. Any other reasons besides time and cost? Accuracy of data, well, experimental methods are always like quite accurate. So if you can do experiments on all of these things, then they can be the most accurate. So typically the computational methods we are trying to develop to make sure they are as accurate as experimental methods, right? So if our benchmark is always the experimental methods. So it's not that they are more accurate than experimental methods. They can be in some cases, but most of the time we're trying to match the experimental accuracy. Enhance efficiency by saving time, by saving cost. That makes sense. We can handle large data sets because they are cheaper, they are faster. So you can handle large data sets. Any other thoughts as to why you want to use these tools? Well, how does it follow ethics? Well, you don't have to kill animals. Maybe that's why you can follow ethics while doing these things. Any other ideas why you want to use these tools? Same thing. They reduce time, they reduce cost. You can screen more molecules in a given amount of time. Less chemicals used, and you also have someone saying that you can predict different hypotheses and you can visualize things. Yeah. Anything else? Any other ideas? Okay. All right. So these are all good suggestions. So let us try to figure out what are the reasons why you want to use these tools. So if you look at the drug discovery process, as I outlined to you before, we have these different stages. Now explaining in more detail here. So you have your R&D phase, you're trying to identify good drug molecules. And so this involves your target ID validation stage, hit ID stage, hit to lead, lead optimization. And then once you have a good molecule, then you go for preclinical trials where you test these things in vitro and in vivo in animal models. Then once you have done the testing, if you have a promising molecule, you file for a IND, which stands for Investigation New Drug. And then once you have that, then you go for clinical trials where you do phase one trials on a few humans to check if your molecule is safe for the people to use. In phase two trials, you see if the molecule can treat the disease or not. In phase three, you do it for more humans or a larger scale, checking both the safety and the efficacy profiles. So the entire time for this process is around 13 years typically, and the cost is $2 billion, which roughly translates to 1,600 crore rupees. If you look at this, all these stages here, the computational tools can help you with all these approaches, these blue colored areas. They don't help you with any of the preclinical or clinical trial stages. These days, people are coming up with artificial cells to mimic your human cell or your animal cell, so you can do the testing in those artificial cells. But synthetic cells, I won't say artificial, they're synthetic cells. But that still requires you to do wet lab work. What the computational tools can help you with this approach here, these stages. Right now, you can see that the cost for doing these things is around roughly $600 million. With these computational tools that have been applied so far, we have reduced the cost to less than $10 million for a given project. And the timeline for doing all these things, including preclinical trials, is right now 1 to 2 years or maybe 3 years at the most. So your chase is from 8 years on average to less than 2 or 3 years, which that this was done because we can handle large data sets and we can be fast, we can be cheap, we can do all of these things in a short amount of time. The other reason why traditionally it used to take such a long time for these processes was that when you came up with the manual design for the molecule, the only way to test the molecule was that you had to synthesize it, and then you had to do the wet lab experiments. So let's say for a given project, you came with 2,000 different design ideas as to what molecules might work for a given protein. Only 10% of these were shown to be binding to the protein. So the remaining 90% of molecules that are synthesized or came with the ideas were not working. So 10% of 2,000 will be 200 potent molecules that you identified. And for these 200 potent molecules, you have to optimize to make sure that they don't have any issues with toxicity, absorption, ADMT properties in general. So what people observed through these different review articles was that only around out of this 200, around 0.41% of the molecules used to have good properties to enter into clinical trials. So 0.5% of 200 will be one molecule, zero point 0.4 is less than one molecule. So quite often your process will completely fail just because you didn't have a good set of molecules to start with. And the reason why they only test a few molecules was because synthesizing these molecules is a challenging task. Because this takes time, you can only evaluate a few molecules that you're interested in, and that limits the number of molecules you can have as potent molecules. and that limits further the number of molecules you can have for a successful drug screening. The other issue with the traditional approach was that, let us say that you found some molecule to be working. You have no idea why it worked. And if some molecule is not working, you have no idea why it doesn't work. You may come with a hypothesis, but they have no idea at the atomic level detail what is happening between the protein and the ligand. So these computational tools help you in not only figuring out if a molecule will work or not, it also gives you an atomic level picture. This is what we're looking for. So they are faster, they're cheaper, but in addition to these two advantages, they also give you this atomic level detail based on which you can develop a rationale as to why a molecule is working or why a molecule is not working. So for example, if you take a look at this video, with these computational tools, you can inspect your protein structure closely. You can see exactly where every atom of the protein is. You can see where the alpha helices are. You can see where the beta strands are for a given protein. You can identify the binding pocket for a given protein. And then you can place your molecules in the binding pocket or dock your molecules in the binding pocket. Each molecule, depending on the kind of chemistry it has or groups that it has, will have a different orientation binding pocket. It will have a different interaction pattern in the binding pocket. So for example, this molecule has this particular shape in the binding pocket, and it also has these interactions will be shown as this dashed lines here. These are the interactions between the ligand and the protein for this ligand and the protein. So as you change some groups on your molecule, the interaction pattern will change, and that will indicate the strength of interaction between the ligand and the protein. So when we use these tools, what we're getting is, in addition to saving time and also saving cost, we get atomic level detail that helps you understand why a molecule works and why it doesn't work. So when you're trying to use these computational tools, always keep in mind that you have to focus on the atomic level detail whenever you run these calculations, and also when you're trying to analyze the results of these calculations. So with those atomic level data, you can try to come up with a rational solution as to why a molecule is working or why it is not working. And we'll spend some more time figuring out how to look at this atomic level detail, how to figure out if a molecule is good or not, and that will be covered in the hands-on sessions later on. So as we addressed, the computational tools are faster and cheaper, and they also provide atomic level detail. So because they are faster and cheaper, before CAD, what people used to do was they used to come with this 2,000 different design ideas, and they used to find 200 potent molecules, and very few of them used to have the best ADMET profiles. If you're lucky, you have a 34% chance of your molecule entering into clinical trials. With the new approaches, it is no longer like CAD, it's no longer computer-aided design. These days, every pharma company or any large company they think about, they use computational tools as the first approach. So it's no longer like computer-aided drug discovery, it's computer-driven drug discovery. And they screen billions of molecules with different computational methods. And then they narrow it down to few thousand molecules, few hundred or few thousand molecules. And because they have started with a large number of molecules, the chances of you finding a good drug molecule is much more greater than one than what you had here. That's the whole idea with computational tools. And while you're doing the screening of large number of molecules, you also get an idea as to why a molecule is working and why it is not working at atomic level detail. So if you want to use these tools to assist drug design, you have to focus on this atomic level detail. So typically, you're going to work with protein complexes. So you have to be aware of the protein structure or the properties of the protein that you're going to typically have. All of you must be aware of these basic concepts, like you have a primary structure for the protein, which is just the sequence of amino acids for that particular protein. And you have to be thorough with the 20 different amino acids that are commonly that are present naturally. Some of them are hydrophobic amino acids, some of them are polar amino acids, hydrophilic amino acids. Some of them are charged amino acids, and some of them have different properties like cysteine or glycine or proline. You have to figure out, you have to be aware of all of these different properties of these amino acids. And these 20 amino acids in a given protein can combine to form different alpha helices, different beta sheets, and these helices and beta sheets can combine to form a tertiary structure. If you have multiple chains in your protein, then these individual chains can combine to form a quaternary structure for your protein. You have to be very thorough with these 20 amino acid properties. So which one's hydrophobic, which one's hydrophilic, because it is very critical to design a good molecule to interact with the protein. The second thing that you have to be aware of is that proteins have different conformations in the active form and the inactive form. For example, if you take a look at the video on the right side, you can see that right now the molecule is in unbound form, and when this pink molecule binds to the protein, let me just play the video again. When this pink molecule binds to the protein, you'll see that the shape of the protein will change. So if you are focusing on atomic level detail, it is very, very important to you to figure out if you want to start with an unbound protein or a bound protein. Because if the position of the protein atoms change, then the atomic level detail will change, and that will change the whole interaction pattern between your molecules and the protein. To give a better illustration of this, if you look at these two structures on the left, you have the active form, you have the inactive form. And you can see that in the inactive form, the two domains are away from each other. In the active form, the two domains come close to each other to interact with this gray molecule. So be aware that the ligand bond structure will be very different from the ligand unbound structure. Most of the time for drug discovery, we can take, well, there are approaches where people have targeted active forms. There are approaches where people target inactive forms, but you have to be thorough and aware as to which form you're going to target and for what purpose. If you want to design a antagonist, you want to target the inactive form. If you want to design an agonist, you want to target the if you design an agonist, you want to target the active form. If you want to design an antagonist, you want to target the inactive form, typically. However, most of these protein molecules will have a common substrate that they bind to. In the case of enzyme, it tries to interact with some substrate to form a product. In the case of a receptor, it also has a substrate which is useful for downstream signaling. So if you take a look at this normal binding process, every protein is going to have some molecule that it binds to do its various functional activities. If you want to inhibit that protein that is causing the issue or the disease that you're interested in, then there are two approaches for you. One, you can try to inhibit the molecule competitively, or you can try to inhibit the molecule non-competitively. In the case of competitive inhibition, the idea is that you want to design an inhibitor that can bind to the same binding pocket as a substrate. And this inhibitor will bind more tightly than the substrate. So if it binds more tightly, then you'll have more of the inhibitor bound proteins compared to substrate bound proteins. And that's how you inhibit the entire protein that is present in the cell. In the case of non-competitive inhibition, or also known as allosteric inhibition, your molecules bind to a different binding pocket on the protein. And this binding event at the different binding pocket will change the shape of the primary binding pocket. And because the shape here changes, the substrate no longer bind to this primary binding pocket. And that's how you inhibit this particular protein non-competitively. Designing non-competitive inhibitors is a bit more challenging than competitive inhibition. But people have done these things, and that requires more expertise. For the workshop, we'll mostly focus on competitive inhibition, but keep in mind that people have also designed non-competitive inhibitors through computational methods. Yeah. So going back to these things, as I told you, it is a time-consuming process, and it takes that long because it's not just about figuring out if your molecule binds to the protein or not. You also have to make sure that your molecule binds only to that protein. It has to be selective to that protein only. It also has to be soluble, it has to be bioavailable, it has to be cleared after a certain amount of time, it has to be permeable, it has to be synthesizable, it should not be toxic also. So in a typical drug discovery project, you have these different properties that you want to optimize, and you have a series of molecules for which you're trying to compare against these properties. And you want to pick the molecule with the best of all these properties. Even if you have like one red area, which one property is not working, it could lead to the entire project being scrapped. So you have to make sure you have the best of all the properties for a given molecule. So what people often do is, in drug discovery, they start with molecules that are efficient to interact with your protein of interest. So you're trying to find hit molecules, which are shown as blue dots here, that are efficient to interact with the protein of your interest. And some of these might be more potent than the other molecules. So these are your typical lead molecules. Once you have a lead molecule, you have to make sure the molecule is also safe, it is stable, it can be absorbed, it can be soluble. It has to have the best of all these properties that are listed above. And these are only some of the properties. There are other properties also that people are often interested in. And you want to find the ideal molecule that has the best of all these properties. So it is a challenging problem. That is why it takes such amount of time, and it's very expensive to do this. And if you have even one property failing, the entire project has to be scrapped and you have to start with a new chemical series of that might work. So it is a high risk area, but if you have a successful molecule, then you also get high rewards for it. So what people do is they first address the issue of potency, and then they go for other property optimizations. So for potency calculations, one of the common things that people often use is structure-based drug design, where the idea is that you have a protein structure available for you, and you want to find if a molecule can bind to the protein tightly or not. So for this, there are two things you have to inspect at atomic level detail. The first thing is your molecule complementing your protein geometrically? I'll explain what that means later. And is your molecule complementing your protein electrostatic or interaction-wise? So people often compare this to lock and key hypothesis, where you have a lock and you want to find the key that fits exactly that particular lock. So you have so many proteins, and so each protein will have its own binding pocket shape, and also the chemistry in the binding pocket might be different. So you're trying to find a molecule that can fit exactly into that binding pocket and also have good interactions with the amino acids in the binding pocket. So for example, what I mean by geometric complementarity is that let's take a look at this particular protein binding pocket, which has a shape that looks like this. Your molecule should have a shape that complements the shape of this binding pocket. For example, if you have a groove here, you need to have a protrusion in your substrate to go and fit in that particular groove. Similarly, if you have one more groove here, you want to make sure that your molecule can go and occupy that groove also. In the other way, if you don't have a groove, that means your molecule should also not have this protrusion to complement the shape of the binding pocket. And for interaction-based complementarity, you're looking at, if I have a hydrophobic amino acid in the binding pocket, then I should have a hydrophobic group on my ligand also to form hydrophobic interaction with the two groups over there. If you have hydrogen bond acceptor in your enzyme binding pocket, then you have hydrogen bond donor on your substrate to interact with the acceptor. If you have a positive charge in your enzyme, you want to have a negative charge to interact with a positive charge. If you have more hydrophobic groups here, then you want to have more hydrophobic groups on your ligand to interact with that. If you have a hydrogen bond donor, you want to have an acceptor on your ligand to interact with that. So you want to complement both the geometry and also the interaction pattern with the protein binding pocket. If you do this properly, then you'll have a good molecule to interact with the protein. So one of the commonly used structure-based drug design approaches is docking. There are other approaches too, which we're going to discuss tomorrow, but today we'll keep with the most basic one, which is docking, molecular docking. In the case of molecular docking, there are two things you are trying to find out. What is the binding mode? And the second is binding affinity. So I showed you this video before. By binding mode, we are talking about the geometric complementarity. So each molecule will have a different pose or mode of binding in the pocket. You're trying to find the right binding mode for each molecule. So you can see that some molecules will bind like this, some will have a different orientation, some are linear, some are bent forward, some are bent backwards. All of these are different orientations of molecules. So docking will try to find out what is the right orientation, the right binding mode for each of these molecules. And the second thing is, for each binding mode, it tries to find out the binding affinity also for these molecules, where it's trying to see what is the strength of interaction for the molecule and which one has the strongest interaction across all my molecules. So you also get some number that tells you which one is the strongest, or which ones are strong interactions compared to weak interacting molecules. So docking essentially tries to find both the geometric complementarity and the interaction complementarity, in this case referred to as binding mode and binding affinity. So how does it do this? How does it try to find the binding mode and binding affinity? So for any docking protocol, there are two different stages. One is known as a search algorithm, and the second thing is known as a scoring function. And what you're trying to find out is that for a given molecule, what is the appropriate binding mode? So to find the right binding mode, it has to generate all possible binding modes for the molecule. The way it does this is, if you take a look at this molecule, you can see that there are quite a few single bonds on this molecule. And these single bonds can be rotated without any penalty. So you can try to just rotate these single bonds here, and docking will generate all these poses for you. You can see that as you rotate around this bond, you'll have different orientations of this aromatic ring, and also different orientations of this entire group here. So docking will generate all these different poses for a given molecule. And for each of these poses, it's going to tell you how tightly the particular pose binds to the protein. So your scoring function that tells you the binding affinity between the ligand and the protein. Okay, so once you have this, for the best pose for a given molecule, you'll have the best binding affinity, and that is given typically as the output. So for the sake of docking calculation, there are four different stages that you have to go through before you can go for the docking itself. You have to prepare the ligand molecules that you're interested in to see how they're going to interact with the protein of your interest. Then you have to prepare the protein structure also. I'll explain what it means to prepare ligands and prepare proteins. Then you have to define the binding pocket of the prepared protein. And finally, you will dock the prepared ligand molecules into the binding pocket of the prepared protein. So keep in mind that every docking process will require these four different stages: prepare ligands, prepare your protein, define the binding pocket, and then dock your molecules. So what is meant by preparing ligands? Typically, when you get structures of ligands from some database, or maybe even if you sketch them, you're sketching them often in 2D format and not in 3D format. So you have to convert your molecules from 2D to 3D. That is one stage of ligand preparation. And then you have to also find stereoisomers for your molecule. You may have, if you have one color center, you can have like one R form, one S form. If you have two color centers, you can have like multiple R forms and S forms of the same molecule. So you have to figure out all possible stereoisomers for your molecule. You can also have tautomers for your molecule. You can also have different charge states for the molecule, depending on the pH of the environment. So for example, in the free solution, when the molecule is not bound to any protein, this particular molecule can exist either in this state or can exist in this state, where you can see that this nitrogen group here has a positive charge on it. So in the case of the free state, like there is no nitrogen, there's no protein of nitrogen here, but in this case you have NH plus here. And this state is not favorable to exist in the free solution, but when it binds to a protein, this positive charge can interact with negative charge amino acid, and this particular state might be more favorable compared to this state here. Even though in solution, the molecule prefers to be in this particular charge state, hydrogen state, it may prefer this orientation in the binding pocket because this gives it a scope to form more interactions than what was present initially. So keep in mind that the ligand can have different charge states depending on the environment. And the ligand, the protein bond conformation of the ligand will be different from the free ligand in solution. So LigPrep is a tool that will take your molecules, convert them to 3D. It will determine all positive stereoisomers for your molecule. It will molecule. It will determine all charge states also for the molecule. And then when you do the docking calculation, you find out which charge state or which tautomer or which stereoisomer is preferred in the binding pocket of the protein. It's not necessary that all the stereoisomers, all the tautomers, all the charge states will have the same interaction pattern with the protein. You can only determine after doing the docking calculation as to which one is the most preferred stereoisomer or the most preferred tautomer or the most preferred charge state for the molecule. Okay. So based on this penalty, if your interaction is stronger than this penalty that we have here, that means this particular form will be more preferred compared to this form, even though this form is the one that's preferred in solution state. Right? This might be a bit complicated, but we'll address this in the hands-on sessions later. Just be aware that LigPrep is a tool that can try to explain to you, that can try to prepare your molecules for docking calculations, where it converts you to a 3D form, find stereoisomers, find tautomers, find different charge states for the molecule at a given pH of your interest. Similar to preparing your ligands, you also have to prepare your protein molecules. The protein data bank is a very good repository for protein structures, but often these protein structures, even from the PDB, will have missing hydrogen atoms. They can have missing loops and side chains. They may also have metals in the binding pocket. For metal proteins, it's very important to make sure you keep the metal in the binding pocket. You may have some other issues also with the protein. So you have to resolve all these issues. And for that, we have a panel called Protein Preparation Workflow that will help you fix all the issues with the protein before you go for your docking calculation. And since we are looking at atomic level detail, it's very important for you to keep the protein structure intact at atomic level detail and also the ligand structure intact at atomic level detail. Once you have done these two things, then you identify the binding pocket on the protein. There are two different methods for this. One is that if you have, so sometimes with the PDB, you also get a protein with a ligand bound to it. So this bond ligand is known as co-crystallized ligand. If you have a co-crystallized ligand bound to given some part of the protein, most likely your binding pocket will also be in that your molecule also bind the same area of the protein. So if you have a co-crystallized ligand and it binds here, you can be sure that your binding pocket will be in this area itself for the protein. If you don't have a ligand bond structure available for you, then you can use a tool called sitemap to predict the binding pocket on your protein. So both of these approaches are available for you. If you have a bond ligand, use that information to define your binding pocket. If you don't have a bond ligand, you can try to use this sitemap tool to predict different binding pockets in your protein. Once you have the binding pockets, then you do the docking calculation. So in the case of docking, you're trying to find the binding mode and the binding affinity. With Glide as a module, so LigPrep is for ligand preparation, protein preparation workflow is for protein preparation. Then you have Glide for doing the ligand docking with Schrodinger. And so it takes around 10 to 20 seconds per molecule with Glide. And we are currently working on a faster version of this Glide, which will be released by July end. So we have a new version of the software released every 3 months. The next version, which is coming in July ending, will have this faster Glide, which will be 60% faster than the timings I'm showing you here. So if it takes 10 to 20 seconds, the new Glide will do it in like 4 to 8 seconds per molecule. So it's really faster than what it is right now. So with these approaches, you can screen thousands of molecules in a few hours. You can screen them. And once you have done this, you'll get a docking result like this, where you have your protein. And then you have these different ligand molecules that can that are docked to the protein. And you can see that they all have different interaction patterns. Some dash lines appear here, some appear somewhere over there. So you want to figure out which molecule will have the best pose and the best interaction. And we'll discuss thoroughly how to interpret these results in the hands-on sessions later. I do want to clarify here that if you read different papers, people often present different ways of trying to shortlist molecules after docking. So this paper published in 2021 did a survey of people from academia and industry, where they tried to compare the different criteria that people often use, and you have all these criteria here, and they gave a score of 1 to 5, 5 being that property is more, that criteria is highly prioritized, and 1 being meaning that that particular criteria has a low priority. So if you look at all these criteria, you can see that the most prioritized one is it similar or analogous to crystal structure? And the second thing that they also look is it complementing the binding pocket in terms of shape and in terms of interactions? Okay, so if you can figure out if your molecule fits exactly in the binding pocket the way it's supposed to, and if it can form good interactions with the molecule the way it's supposed to, then you'll have a very high chance of the molecule being a good molecule. So this, to inspect the geometric and the interaction complementarity, you have to look at the atomic level detail as to how each molecule binds to a given protein. And since you can only do such kind of analysis for maybe a few hundred or 1,000 molecules, and you can't do it for lakhs of molecules, people use docking score, which is a least prioritized approach, to filter from lakhs of molecules to a few hundred molecules based on docking score. And for those few hundred, you have to spend the time looking at the complementarity of your molecule with the binding pocket in terms of shape and also in terms of interactions. So if you do this correctly, you're going to have a good molecule that you can interact with, that you can take forward for wet lab studies. So keep in mind that docking is not just for small molecules. You can use docking for other kinds of chemistry also. For example, if you want to dock peptides, macrocycles, you can do these tools as well. You can also go for covalent docking if you want to design a covalent inhibitor. We also support induced fit docking. You also support cross docking. In the case of cross docking, you take typically docking is for like one protein, multiple ligands. In the case of cross docking, you take one ligand and screen across multiple proteins to make sure your molecule of interest is selective to the protein of your interest. So with this, I will stop the presentation. The key take-home message is that these tools are faster, they're cheaper, but they also provide you atomic level detail of the interaction pattern between your ligand and the protein. And that is very important for your um, for figuring out why a molecule is working and why it is not working. And once you have this rational approach for it, then you can go ahead and try to design better molecules based on this intuition that you have gained through the atomic level detail. With that, I'll stop this presentation. If you have any questions, please let me know. I see that there are two questions. What do you mean penalty? I hope I explained that, Dushyant. I was talking about state penalty where the molecule in the free solution, in aqueous solution, it prefers to be in the deprotonated form compared to being in the protonated form. But the protonated form might be more favored when it binds to the protein. So if you're focusing on drug discovery, where your focus is on trying to inhibit the protein, then you want to take the form that is more likely to bind to the protein, even if it is having a higher penalty to exist in the free solution. So you have a molecule before it binds to the protein, you have a molecule after it binds to the protein. You have to see the molecule that which form is preferred once it binds to the protein. The level of energy in binding will be explained in the Hansen sessions later. So we will be patient. We're going to cover that more thoroughly in later sessions today. Any other questions on this presentation? As I told you, for a non-binding protein, you mean to say off-target proteins, it should be as low as possible. Like you want to be typically, it should be like -3, -4, or -5, not higher than that. It can't be -6. Yeah, -6 is like maybe okay, but it can't be like -8, -10, and things like that. So we look at what the penalty score represents. Try to ask this question again when you go for liquid, right? And how to select the gen polymers and stuff, we'll also address that in the hands-on sessions later. Okay. The penalty score for the ligand typically in this case is that if you calculate your different functional groups on your molecule, they all have a pKa. The pKa indicates whether the molecule prefers to be protonated or deprotonated in that particular at that pH level. And if it is going to be protonated or if it prefers to be deprotonated, the other form will have a penalty with it because it's not the most stable form at that pH. That is what the penalty here indicates. You don't use, it can be performed, docking won't complain. You can dock even like a very big molecule to some protein. But nanoparticles, you have to keep in mind that they typically have a diameter of typical of around 3 to 4 nanometers, unless you're very good at synthetic chemistry and you can get a 1 nanometer nanoparticle. If you have a 3 to 4 nanometer nanoparticle, then that is much bigger than your protein. So how can you dock a molecule that is much bigger than a protein onto your protein? It won't fit in that way. So you have, even though software won't complain, you have to be careful as to what you dock. If you want to figure out how a nanoparticle interacts with the protein, there are other approaches that you can use, but docking is not the right approach for that. So if you, which software to use is up to you. You have to read. It's not a question of whether you want to prefer online or offline software. It's a question of which one is more accurate. There are plenty of papers that have been published comparing different docking softwares. You can read those papers to see which ones are more accurate and use the one that is more accurate more accurate software, sorry. Please explain co-crystal formation. That is, can you elaborate more on that? What do you mean co-crystal formation? There's a purely experimental approach where people do extra diffraction. They have to get a crystal structure for this complex, for the ligand bond to the protein. And that can take easily 1 month to a year or multiple years for a single molecule. And that's a purely experimental approach. And once you get the structure is deposited in the protein data bank, and you can use those structures in protein data bank for a starting point for drug discovery. But how the co-crystal forms is a complicated question that you have to ask mostly experimentalists. I don't have the answer for that. Any other questions? Yes, glide works in metals also. So if you have organometallic complexes, then you can try to take those and dock them to the protein. It does contain parameters for, I think, six different metals, but if you have something outside the six different metals, we also provide a tool called Jaguar along with the software. And the Jaguar can do quantum mechanics calculations, which can be used to determine the charges on your metal and also the nearby coordinating ligands. And those output of Jaguar can be given as input for docking. So you can dock any metal, any metallic complex into your protein. You can check protein targets with single ligand. So if you're asking about like, can you try to see which molecule, which protein your molecule binds to? That is a much complicated question. If you have a list of proteins that you want to target and you want to see which one it can bind to, then that's more easier. But trying to screen a ligand against all the proteins is not possible because you don't have the protein structure for all these molecules, uh, for all these proteins, and you also don't even if your protein structure, trying to get the right binding pocket is so difficult. So typically, you can screen, in principle, you can screen one molecule against any proteins of your interest, provided you have the protein structure for that particular protein. How does molecular compatibility help structure-based drug design? As I told you, you have to focus on geometric complementarity and interaction complementarity. So if you have the right fit in the binding pocket, that means you have the right key essentially to fit into the grooves out there, right? So once you have the right fit, then you have to look for the interactions. And as I told you, depending on which amino acid in your binding pocket, if you can have similar groups in your molecule present in similar areas of the binding pocket, then you have very high chance of forming good interactions. So you can't have a hydrophilic group near a hydrophobic region in your binding pocket because hydrophobic and hydrophilic don't go well with each other. So you have to have hydrophobic with hydrophobic, you have to have hydrophilic with hydrophilic. That's when you can form the highest number of interactions. And the more interactions you have, the stronger your binding will be. And that's how you can figure out molecular compatibility. And that can help you in structure-based drug design. Any other questions? You have to be thorough with the 20 amino acids. Depending on their properties, your whole small molecule design will be affected. You have to make sure you have good interactions with those 20 amino acids, or some of them in the binding pocket. And if you can have good interactions, provided your molecule fits within the binding pocket, then you have a very high chance of a good molecule with you. If there are no more questions, please, there are plenty of libraries that are available online, which we'll discuss in the hands-on session later. Any other questions? Thank you. Some of these concepts will be new to all of you. I would recommend that once this workshop is over, please go through the recording again. We'll share the recordings with you at the end of tomorrow's session. And that will clarify a lot of concepts that you may have heard for the first time. And the best way to further go into these concepts is to read papers or do your own research and read as many papers as you can to figure out what people are saying. And that will explain to you these concepts even more thoroughly. If there are no more questions, then if anyone has issues logging into cloud instances, please let me know. Whoever has issues with logging cloud instances, please let me know. Can you share your screen, Emmanuel? Can you share your screen? Your password, no, that's not user temp, it's not a username. Username will be your, I gave you username. I think for you, it is user117. Check your email thoroughly. That one tells you it's user117, that's a password. You have to give the whole password. Okay? It's not 117. It's not temp, it's just user117. User dash 117. And then the password is just below that. Each of you have your own individual instances. Please don't try to use someone else's instances. Click on login. And once you get the page, then you click on the second option, virtual workstation. Click on the second option, virtual workstation. And if you get a pop-up on the top left for copying clipboard information, just click accept. But for now, you're good. I think it should work for you. Anyone else has issues with logging into cloud instances? Anyone else with issues for logging cloud instances? We'll start the hands-on sessions at 11 o'clock. So you're going to take a quick break for a couple of minutes, go and come back. If anyone else needs help with logging into the cloud, please let me know. Can you share your screen, Pankaj? Can you please share your screen, Pankaj?
Hello. Hello.
Yeah, we can hear you. Can you share your screen?
But it is showing invalid login.
Can you please share the screen so I can see why you're showing invalid login? There's a share screen option on Zoom. You can click on the share button and share your screen. You saw how Emmanuel did it with the share screen option. Please try to share your screen so we can help you with that aspect. Give the password. The password is in the email. So go to your email first. Please go to your email. It's in the same browser, second tab. Can you hear me? You see on the top, you have the second tab, right?
Password is 6.
No, you have to give the entire thing, Schrodinger dash 6, whatever. Okay. So you have to type Schrodinger dash that thing. So the entire thing is a password. It's not just the last 6 digits.
Okay.
I would recommend you to just copy paste the password from the email instead of typing it. So your email is in the second tab. Just go to the email, copy the password and paste it over there. No. That's what I'm telling you. The password is the entire thing, Schrodinger dash 66, the 6-digit number. So the password will be this entire thing. You can't just take the last 6 parts over there.
Sir, including dash or without dash?
Type Schrodinger first. Type Schrodinger all small. SCH. The password is SCH. SCH. RODI. Can you show me your email first? Can you show me the email with the username and password for you?
Sir, I have received password 86 small q.
No. That's what I'm telling you. Show me the email once. Show me the email with the password. You got the email right with the password details. Go to the email first. Can you please go to the email, which is the second tab in this browser? Can you please share the screen again? Okay, I'll type your password in the chat box. You have to type the whole thing. Just give me 1 minute. So your password, I'm sharing it with the chat box. Okay. Copy and paste the entire thing in the password section. I'm putting the chat box of Zoom. Just 1 minute.
Okay.
Yeah. So you type the password, right? That whole password, the Schrodinger dash 63, you have to type everything. You can't just type 6236.
Okay.
Okay.
Thank you.
Yeah. And don't share password to everyone. Okay. Let me see if I can delete that thing. Yeah. So type that entire password and see if it's working or not. User 70, can you please share the screen? Divyanshi, can you please share the screen? Yeah, go to the software, go to the interface, go to the virtual workstation on the top. Yeah, click on that link. Give your username. You have a space in front, remove the space. Don't copy the space in front. Copy without the space. Yeah, exactly that password. Do the same thing for the user also. User also you put like an extra space in front. There's a space in front of, okay, great, it works. Now click on the second option. Click on the second link, virtual workstation. Close other tab, keep only one tab open. You have two virtual workstation open, keep only one open. Tab 2 and 3 both are virtual workstation, keep only one of them open. Anyone else having issues logging in? If there are no issues, I would like to ask my colleague, Dr. Vinod, to take over for the hands-on sessions. And we'll first go through the Maestro interface. We're going to figure out how to sketch like simple molecules and also look at protein structures and DNA and other things. Then we will go to the calculations for protein prep, lick prep, and grid generation or binding pocket definition. And then we'll do docking and docking analysis. So we'll have a lunch break from 1 to 2 p.m. We'll cover whatever we can till 1 p.m. And then anything else will be done after 2 p.m. Over to you, Vinod.
Yeah, thank you. Thank you, Kaushik, for a wonderful presentation. I will get started. I just want to also one more time check in case if any of you are facing login issues, please let us know in the next, like during the break sessions. Okay. So the break sessions are purely dedicated so that you can try to do. So the core concept is to motivate you to learn the software much easily. Okay. So hope once you have done the login, Let me introduce this operating system as Linux, and all the input and output files for the workshop is well covered inside this data folder. I think my screen is visible and I'm audible. So you could see all the files are available, so you don't have to panic that you may miss out something. All will ensure that you are on the right path, provided you focus on, okay, what things are discussed and how to learn new tools and techniques, how to apply them in your project. That should be the primary focus. Along with that, we also have tutorial videos. We also have step-by-step brochures, so which will be quite easy. So just relax and watch everything now. So if you see all the computational chemistry or computational biology research is majorly done by using Mastro interface. There is a biologics interface also separately and material science suite also. But Mastro is quite famous, and this interface is the visualizer is free for academics. So if anybody likes it very much, I request you, I encourage you so that you can install it in your own laptop and use it for teaching, for visualizing different molecules. Okay, so I will be walking you through the major aspects of the software. So if you notice, when I joined Schrodinger, there were so many softwares. Okay, there were so many softwares, and I was very curious to like try to solve everything at once. So that is, then I realized it is quite difficult technique because the science is quite vast, more than 20 years, and okay, so we will try to focus on that aspect. But if you have any suggestions or have any improvisation, please let me know. Okay, so what is the task? The first task that you should focus on is in the project management. Okay, where you save and where the output file goes. Okay, as I already pointed out that all the softwares are available in the task browse, right? So if I run something, if I do structural alignment, how I will browse the input file and where the output file will go. Okay, so that is the important thing. So for that, in project management category, the first thing that you should focus on is changing the working directory, which is actually accessible from File. File has this option of change working directory. So change working directory is going to resolve all your problems where you will be having a location of your folder. It's a folder, okay? So if I click change working directory, it is coming to my home directory. So I'm using user 131, right? So this is the option right now. You have to create a new folder. So this is day one. Okay, so I have created a new folder. Creating a new folder is like right click, you can create a new folder, or here also you have an option for creating a new folder. Once I do backspace, okay, sorry, hold on, user 131. So here is, you can rename this folder. So this is day one demo. So this is a folder. Please note that whenever you give file namings, don't use space or comma and special character. You can use underscores or colon, but try to avoid blank spaces, okay? Which is very, it create a lot of bugs and issues. So here I have clicked and I have changed my working directory. So this signifies that if I go to task and if I do any run, if I do molecular docking, automatically it is going to go to that particular directory. The second thing, which is very, very important, is to save the project. Suddenly, there is a possibility that you can get disconnected or you are having some issues, you want to close down. And in that case, you can come and reopen this project. So project is a file, okay? So it has an extension of .prg file. So it's like our doc or docs file. If you click file and second thing, save project as. So here it is, which I'm going to do is docking. So imagine this is a docking project and I will save this. So you can give any naming convention as per your preference. So I've done two things in project management. Okay. So first thing is file, change working directory. And second thing is file, save project as. So I request you just give a try to do these two tasks and we are there to support you. But try once I think I can see Debelina has written that. Is it still the problem is there, Debelina? You're having issue? We'll look into that. Don't worry. Okay. So in the meantime, I request others to do this two tasks. Okay. So two tasks. The first task is change the working directory and save the project. So this two tasks I request you to do. Once it is done, just type done. Okay. And after a few minutes, I'll try to help. Thank you, Nafis. I think you have done it. Thank you. Thank you. Yes, Develina, I think you are the one who are having issue, right? So can you please share your screen? Is the username only 3 letters? No. Could you please share your screen? Thank you all for confirming that it is done. Develina, are you there? Anushka, Richa, thank you, Anjali, everyone. I'm just checking if Develina, you're having issue. I too not able to log in saying invalid password. This is the username, only 3 letters. Could you please share your screen? Oh shit. Okay, so you have shared to him, is it? Or Kaushik?
No, I'm just sharing it with you, so in case she has any issues, you can help her out.
Okay, great. Okay,
Unless she shares the screen, we can't help her, right? So we have to know
yeah,
what she's.
correct. Okay, let me check.
The username is not 3 letters, it is user dash 104 for you. And the password is stored in your dash the 6 digits. You have to type the entire thing stored in your dash 6 digits. And the username is user dash 104, not just 3 letters. You have to give the user USER dash 104.
Yes, because I'm not getting response from Devilina. Is it she there? I'm just checking. Yeah, she's there, but
You can, if you need help, you can share this.
okay, we'll try again. Yes. Okay. Thank you, Develina. Just give a try. I can wait for another 2 minutes so that you also pop up. I can see I greatly appreciate all of you. Most of you have done it, and thanks for typing done. That motivates me a lot because to know like whether I'm going in the right direction or not. Okay. So Develina, for Develina, like I think I will wait for another 2 minutes and see if you can do login. Anybody else? So still don't hesitate. We are there to support you only. So in case if you face any difficulty, you can just type here. Swati, thank you, Swati, for doing it. So, okay, Devilina. I think I'll continue and again I will check again, okay, after this part. Okay. Thank you all. I think that was quite easy. I can see the crowd enthusiasm is good. So I will move on. Now, what is the next task? Okay. The next task is imagine if somebody asked you that, okay, could you please explain me a biomolecule? So which biomolecule will you prefer? Or which is the molecule that you like the most? Like, I think it is quite common. But yeah, just I'm checking again. So how many of you like DNA molecule to be like a nucleic acid molecule is quite a fantasy, right? Many of us loves it. So we will be discussing how to sketch a DNA, okay, at atomic level precision, right? Okay. So for that, I think my screen is visible. So you have to sketch a DNA that is available in this build option. I hope everybody can see that. See, the task is clear. We have to sketch a DNA and scientifically evaluate DNA properties. Okay, that is the major task now we have. Okay, so imagine you are looking for 20 base pair of DNA. Okay, so here in the build option, okay, in build option, anything you see of DNA, no, I don't see anything on DNA. There is something called as add fragments. Okay, this is what I will do if the task is given to me. Okay, yes, it seems there is nothing. Okay, fine. That is fine. And but there is nothing of DNA. There is more options here, right? Okay. There is organic is there. There are so many things. Okay, that's great. So like that, you have to explore. But for DNA, we have specially made a very user-friendly interface. That is, that option is available in other edits. Okay. So out of this two, which option will be off? See, build biopolymer from sequence. So this is what I will use. I'll go for build biopolymer, and here is the option. Okay. So I encourage all the students, please read what is there in the panel, right? So this says build biopolymer from sequence, and biopolymer type is peptide. So did I came here to build a peptide? No, I came here to analyze my DNA of my interest. Okay. So DNA has been mentioned here. The grow direction is right now, if you want to activate, by default, it is from 5 to 3. Okay. But you can grow it from 3 to 5 if you activate this. And by default, it is double standard. Okay. So if you type here the sequences, then automatically the counterpart of the base pair will be there. So first thing what you have to do is you have to add the title, right? So I will type here DNA molecule, okay, whatever you want. So here it is a title, okay, here it's not the base pair. Then here you will add A, C, T, G, C, 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 T, 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 A. So only 4 letters are used, okay, A, C, G, T. So these are the 4 base pairs that you can use. At least you type at least randomly for at least more than 20. Is it fine? So if you do that and just click build, that means we have successfully sketched the double helical DNA molecule. Okay, so let's see how many of you are able to do this. Again, I'm giving you clues. It's not an SOP, it's a scientific research software. Objective is to build a DNA, and the panel which you will use here is other edits and build a polymer, biopolymer from sequence. Okay, so I'll give you like, see, please give yourself some time and ask all these questions like I did, and then I'm sure you will be able to reach here. So, and before asking someone, you should try by yourself first. Okay, and if you fail, we are there. I will address all the questions by 11:20. Okay, so 2 minutes, I will just give yourself the opportunity to do it by yourself. Oh, you have done it. So I can see one of you have done. So let me just type here DNA sequence, DNA. So I've typed here. Okay, so I know I keep a track of it. How many of you sketched it? I can see many of you have done. I know it's very basic, right? Okay, Saloni has done it. I greatly appreciate it. Anybody left behind or feel it is quite difficult to do, please let me know. Okay. So don't be in a hurry. Those who are doing it fast, you don't have to worry about them. Do it calmly, think about it. And then once we have sketched this DNA, we will try to scientifically evaluate this. Okay. So I'll have a dedicated session on that. Yes, Nishita. So Nishita, see here, the task is simple. You have to build a DNA and the panel is in front of you. My screen is shared. To build a biopolymer from sequence. Now you read this panel and make it out what you should do. Just give a try. If you still fail, let me know. Share your screen and then I will help you. Can we take? Yes, Dr. Shekha. You can take any sequence, but base pairs only. Okay. Nucleic acid, DNA base pairs. Okay, yes, Develina, I think you need help. Okay, please share your screen. Okay, there is A type, B type, DNA, Z type also is there. So that is a subtype of it. Okay, so Ajanya, yes, Develina, you have to share your screen and I'll help you. Anybody else? Please wait. We will be discussing on how to analyze the DNA, what properties to look for. Everything we will discuss. Okay. So hold on. I think I'm waiting for Deblina. You have to share your screen. I don't know why you are not able to log in. Thank you, Ajay. And thank you, Radhika. Dr. Johi, thank you for finishing. I know this is quite basic macromolecule, but DNA is quite attached to everyone. Deblina, you have to respond. Did you try to share your screen? You are not able to share your screen also. Hmm. So if you can't share the screen, then it will be difficult for me to support you.
May I say something here, Kaushik and the organizers?
Yes.
This is Dr. Gitanjali Yadav. I'm the coordinator from the National Institute of Plant Genome Research. So first of all, thank you very much for coordinating this for our, students. But in case somebody is not able to follow or listen to you, then you can please go ahead. I would suggest please go ahead. Deblina had come at the last minute to apply for this, and if she doesn't want to continue this workshop, she can skip off. But I think others who are there and who have done, it is very important that we are able to continue. So I will talk to Deblina separately in case she's not able to follow what you're doing, but let's move on.
Yeah. Okay. Yes. Yes, ma'am. I greatly value that. I was just giving a chance. So I was
Yes, no,
just
I
checking.
realize how much you are spending time waiting for each individual. That is I appreciate it very much. But there are also a huge number of PhD students. Deblina is not our PhD student either. So we had added her at the last minute. And I realized that possibly there is some glitch in the connection. But please carry on for the other students who are following you.
Okay. Yes. Any
Thank
students who feel left behind, don't worry. The recording is on, and Kaushik will be sharing it later. But thanks for your inputs, ma'am.
Yes.
So I will be sharing now the screen, and I'll continue because we have a very beautiful DNA in our screen. Now it's our time, so please, I request you close the builder panels, and right now you may have something like this, right? Now you are curious to see this, what exactly it is. And this part of the session, we will be focusing on how to control this structure, how to move this. Okay, now options and clues are available in view. So you could see here there is something called as K, J. So try this, press K continuously and then J continuously. You would notice that the K and J is helping you to zoom in and zoom out. Okay. And I'm going to go and check again in the view section. There is options for rotating. So you have colon, semicolon, close bracket, okay, open bracket, close bracket, all those options. So if you do that, then it will help you to translate the structure, okay, in different orientations. Sorry, rotate the structure and translate the structures. So I request you, please at least devote some of your time. That is, you can see here, KJ, focus on some of the rotations options in keyboard and then do translations. Okay. And some handy shortcuts which is going to be useful is Z. Z is a single shortcut which is going to fit the screen. Okay. Wherever if you are lost, like suddenly DNA is not visible, click Z, you will be able to see that. Okay. So please focus yourself on rotating the structure, zooming in, zooming out, and translating the structure. The next thing you should see in the macromolecule is to focus on window and sequence viewer. So if you see in the sequence viewer, you will be able to see the base pair informations. If you activate, see here, the base pair informations are visible. And now you can click them and click Z, then it will focus on to that particular residue, a base pair of interest. Okay. So if I click here, click Z, it is going to focus on that. In case you want to deselect, you can deselect by clicking in an empty space, or you could see here, there is like, for example, if I selected here, see it is going to have this all and deselect option. But usually I deselect by clicking in this black screen. If I do that, it get deselected. Click Z, then you should see the entire molecule in the screen. So this is on how to select, and usually the selection of atoms is done by, in this panel, you can do residue-based selection, or you can do atom-based or molecule-based. So we can do chain-based also. So if I do like click on single chain, a single chain is selected. Okay. So like that, you all have to focus on rotating, translating the structure, and zooming in, zooming out. Activate the sequence viewer, do selection and deselection. I hope for this section. But to add little spice on this, I request you to please activate this style, activate the ribbon, okay, and activate the surface. Okay, so this is what I expect everyone to do so that you get more information on the DNA molecule. Okay, so that is you are able to see the major groove and minor groove. And already I showed you how to zoom in, zoom out, do translations, okay, and rotations. And I told you how to activate this sequence viewer that is there in the view. Okay, sequence viewer, do selection and deselection. I hope this is what I expect right now. So I will type it, and if you're done with all this, okay, then we will proceed further. Okay, let me just type. So this section is dedicated for DNA rotations, translations, uh, and then you have to activate the ribbon and surface view, okay, from style panel, okay, and then you have to okay, and then do selection and deselections. I have not told you how to control the structure by using mouse. That also I will show you now soon. Okay, so just take yourself some time, play around with this DNA, and let me know. Okay, after 2-3 minutes, I will check if anybody have any questions. And if you have successfully done all this, and you have to activate the sequence viewer also. Okay, so which is in the window option. Okay, yeah. Thank you, Jyotsna. I think that was quite quick. Okay. So in case those who are trying to do it by mouse, if you scroll, it will zoom in, zoom out. And if you press the center click, then you can rotate in 3D. Just do the movements in mouse also, it will work. Okay. So this is quite crucial. I will dedicate 5 minutes. I will wait and I'll try to help everyone. Let's see if all of you are comfortable in analyzing your DNA of interest or not. See, focus on identifying options for DNA rotation translation. Activate the ribbon view and surface view from the style options above. And then you have to play around with selection and deselections on the DNA. If you're confident on all these aspects, that's great. Otherwise, I'll help you after 2 minutes. Okay, so yes, I think if you want to rotate it using mouse, do a center click. That is the cursor, right? Where you scroll. If you scroll the center wheel, if you press that, then you will be able to do the movement in 3D. Okay, so let me just reshare it. So this time, this is purely dedicated for moving the structure using mouse. Okay, so if I keep the cursor and scroll, it will zoom in and zoom out. And, and then if I do a central click on the wheel, then it will move in 3D. Okay. Now, some of you may have different kinds of mouse and some of you may have exposure with PyMol. So in that case, I encourage you to customize your mouse actions using whatever the mouse you are using. And if you don't have mouse, use this option of PyMol mode or trackpad mode. Give a try and get comfortable in moving the structure. Again, I'm repeating the options for customizing the mouse actions are there in the workspace, customize mouse actions. Okay. So just give a try and then we will see. Okay. Is it fine? Yes, Saujanya. So if you seeing the ribbons, okay, you can activate from style option. Okay, here. And if you see, there is an option for activating the ribbon and activating the surface. And this can be deactivated also in the bottom right, okay, which is like this. If you click, it will deactivate. You can remove the sequence viewer also if you feel like that purpose is not required. So if you click, then you can deactivate. You can activate surface and ribbons also as you do. Okay, Shikha ma'am, like I think you see here. So I will share you again. If you notice in workspace, there is a customized mouse action is there. And in customized mouse action, we have to change this to trackpad mode or PyMol mode. Is this okay? Thank you. Anybody else? Just play around. Please get a feel about your DNA. Anjali ma'am, like I think connecting with the server. If the internet is not stable, then it will be difficult. Check for your internet speed, or because I think everybody else is able to do, or you change the network if it is possible by connecting through a mobile, or if you have other hotspot, then use that. Okay. Thank you, Lavanya, Saujanya, everybody. So I'm just checking if anybody still have problems. Anybody need help or any questions, or shall I move forward? Translation, Rakesh sir, I just told you, right? All options are in front of you here in view. So you could do translation by using question mark, colon, a curly bracket and all those things. I can't rotate. By using keyboard, then I request you to configure your customized mouse actions and then able to, okay, do that. Thank you. Just change this option, okay, then you will be able to do whatever you want. Yes, it's Momal, I think Momal, right? You can't rotate. Can you try just center click? Yes. So can you do a center click and try to rotate it? It will move like this. XY will come. Then you can move in 3D. It isn't. Okay, please share your screen so that we can check what's going wrong. Okay, did you press dot in the keyboard? Is it not working? Dot continuously. Okay, yeah, see it's working, right? Then semicolon, question mark. Yeah, and mouse or no mouse? Mouse is there, then go to workspace and then customize mouse actions. And then you want to change it to Pymol mode, just give a try and close it. Yeah, close this. Okay, you want to Tanya. Okay, I'll come back to you, Tanya. Okay, so now you try. You're able to do, if you do right click, left click, it will do some translation. Just try it out. No, no, left click or right click if you do, then you should be able to see that. No, you're doing now. So it should work. No, you should not click on that is a keyboard shortcut. Maybe it is not allowing your keyboard input. So that could be the reason. So that's the reason I'm just checking if you can use your mouse. But I think the other shortcuts are working, right? Comma, question mark, curly brackets, and all those things. Can you try to rotate it for the time being? Okay, so just give a try. See, this is minor, I guess. I think you will figure it out as we go along. Okay. So Tanya, like unable to change style. What style do you want? How to remove surface? So I think that also I showed you, right? It's in the bottom right, the toggle option. This is not removing, this is hiding the surface. So here is hiding the ribbons. Okay. So you can activate and deactivate. Usually here is the surface symbol, which S with blue color S, which suggests the surface has generated. Okay, so if you click manage, then you will be able to delete it also if you don't want. And to generate, you can click on style and activate the surface. Okay, is it fine? So what else? Okay, so I'm just checking for the last 1 minute. If anybody still have issues in sketching DNA and controlling the structure in 3D workspace, then selecting, deselecting. Please show again how to select. Okay, so Nafis, right? If you see here, if you want to select complete chain, okay. So I have to change this to chain, okay, chain mode, and just go and select. Then it will select the whole chain. Is it okay, Nafis? Yeah. Anybody else? So have you finished all the things which I have shown you? So shall I proceed? I will wait for another 10 seconds and then I will continue. Are there any other questions? How to select full DNA? Just do Control A also. That's nice. If you do Control A, it will select the full and then you can deselect if you want. Anubhav Sahu. Yes, that's a good question. Anybody else have any other questions? Otherwise, I'm moving forward. So what else you want in a DNA? Did I show you everything? I showed you the grooves. I showed you the double helical DNA view, but I did not show you the non-bonding interaction that drives. I'm able to see left side panel workspace. I'm not getting what you're saying. You are not able to see. Okay, not able to see. So this could be the reason that because you have closed this. Okay, so that you can activate. Any window can be activated from here. So this is the workspace navigator option. So if you click this, then you can see again. Is it okay? Mamta? Okay, great. So what else you want to see in a DNA? The major driving force for this helical structure is the non-bonding interaction. So the non-bonding interactions are in the bottom right. So if you see here, the non-bonding interactions can be activated and deactivated. So, but right now it's not showing anything, right? If you focus, if I activate, nothing is coming. Why is it so? The reason is by default mode, it is for ligand receptor, okay? But we can make it all, and if you click all, then you will be able to see everything, okay? That is contact clashes. So I request you to activate all the interactions, like aromatic hydrogen bonds are there. There is, okay, there is this good interactions are there, okay? So you can activate and deactivate every interaction and get a feel about it. Okay, so C aromatic hydrogen bonds are there, halogen bonds are there. There is pi cation interactions are there. There are contacts which is actually good, bad, and ugly. Why you have this many good interactions? So don't you feel this is nothing but hydrophobic interactions? And you can see this much that also is in a shape of helical view. So majorly the non-bonding interactions are the core reasons for this double helical structure. So you can activate and deactivate as you go like this. Okay. So please play around and I request you to go through all these non-bonding interactions. Okay. Non-bonding interactions are very, very important to visualize them and get a feel about the DNA. Okay. So DNA by default, it will not come because it is in the ligand receptor mode. So if anybody have any troubles, first you try and then reach out to me. Okay. Thank you. Yes, okay, let me just show it again. Yes, let me just see here. So you can activate and deactivate the interactions in the bottom right. Okay. If you do right click, then you can see all the interactions and make sure you activate this all option. Okay. All, and in the bottom also all, and in the bottom most also you have to make it all. So if you do that, you will be able to see all these interactions. And I request you to activate and deactivate all the individual interaction and get a feel about it. So like this, okay, you can hide it. For example, I'm just seeing now hydrogen bonds or non-covalent bonds. This is pi interactions, and these are the contacts. Is it okay? Thank you. So please keep in mind, it's not about me trying to like, it's about how you should also explore, because there are a lot of softwares, lot of options. Not every time will be there someone to help you. So as I said, this is for, I'm trying to motivate you how to do the self-learning. What is my task? The task is to explore the DNA, and I have to look into every. Okay, fine. So shall I move forward? Is it like everybody has done this? Or if still anybody has any issues? Okay, that's great. Fine. So now what we will do right click and clear the workspace. Please, everyone, right click and clear the workspace. What is the next topic which you feel like we should discuss? Man, I have to check if all the participants are. I have to check if my has poll options. Let me just see here. Participants. Okay. So how many of your came is I have to see here. There should be polls. Yes, I have polls. I think maybe he has not changed it. Yeah. So I'm going to launch you a poll. I'm sure. Please answer this so that I get to know the next topic very easily. Can you please answer these questions, which is on your screen now? Okay, so I see around 68 of you have responded. I think this is sufficient. Maybe just I will share the screen for the major participants. So some of you are advanced also. That's nice. Okay, so please, I think all of you can see the results, the poll results, I think. So you can see the first question is, please share your experience with computational biology or chemistry. So some of you are, I think 9% is a quite good number, but most of you are medium, basic, and freshers. For the advanced and medium, if you have like little in-depth questions, I request you to reach out to us later and we are open for discussing. Okay, but this workshop is purely basic, so there are not much advanced things until unless you ask for it. So did you have earlier experience? Some of you have said yes, that's nice. And what about the broad? See, I can see many of you are biologists and some of you are from other fields. Okay, that's great. Let me just stop the sharing. Okay, fine. So we will now dive into sketching a small molecule. Okay.
Just one small thing for
Yeah.
those who are advanced also. Tomorrow morning's presentation will be quite useful for you, so please join for that. If you think this is too basic, tomorrow presentation will be covering a wide range of topics. Thank you.
Yeah, sure. Yes, in the presentation, Kaushik will be covering many advanced application also. So please don't miss out on the morning presentation. Okay. So if you see here, I have done DNA sketching. Now, what about small molecules? Small molecules are also interest. Now, the task is to sketch a small molecule using 2D Sketcher and 3D Sketcher for derivatives, okay, for creating derivatives. Now, that you can quickly do it by using edit option. And in edit, there is a 2D Sketcher. So I'm sure many of you have used ChemSketch, ChemDraw, and all that. So I request you to do two things, one attachment and one fusion. Okay. So here is one benzene ring I have placed and I will attach a fragment of my interest and then I will fuse another ring. Okay, let's see here. Yeah, so I'm going to drag here, see here, a methyl group I have attached and I'm going to fuse the 5-membered ring to the 6-membered ring by clicking on the bond. Okay, a small sketching, it's a nice experience. There is a very high chances that you may make some mistake like I have done it. Now you can delete it by using this option, this eraser option. Okay, so the concept of sketching is attaching, fusing, and anything you want to add some hetero group, that's up to you. Okay,
Sir, can you please repeat this?
which part?
This 2D sketcher part.
Okay, so you should be able to get the 2D sketcher. If you click edit, there is a 2D sketcher and you will be able to take any fragment of your choice from the left side panel. And here I'm going to delete everything. I will do again. So I request you during sketching, I want you to focus on two things. That is attaching a fragment, methyl ring. Okay. And then you have to, hold on, I think I did a mistake. So you can do Control Z also if you're doing mistakes. So it will go back. And if you click here, you can drag it and it will come. Okay. Let me just see. Yeah. So here you go. So there is a methyl group. And fuse a ring, another ring by clicking on the bond of it. Just take a 5-membered ring and then click on the bond, then it will fuse. Okay, and you can add different hetero groups as per your choice. Please consider the valencies of the structure. So let me just see this. Yeah, so I have added it here. And once you have done this sketching, and in case if you do some mistake, here is the delete options. Like if I just place something wrong, then I can delete it here also. So you can delete it, click save as new, and this is S2 and click OK. So in the bottom, save as new option is there. Once you are done, you will be able to see a structure. See, currently it's going to show some interactions. You have to switch off your interactions if you are activated it. Then you are all set to see the 3D molecule. And this molecule also you can control it as per your choice. Okay. The same 2D sketched molecule, you can try to do a 3D attachment. Okay. So that you can do it by clicking, but see here the issue is it's selecting the whole molecule. For doing a 3D sketching, make sure the atom mode is on. Okay. The atom mode should be on. And you have to, for attaching, you have to click in the terminal atom. Okay. And here also, I'm just going to build and going to add any one functional group, okay, which I've added. So I've attached a amine group. Similarly, I can fuse a ring by selecting two bonds, okay, two atoms. And if I go to build, and then I can again fuse ring into this. So you notice, and if I do mistake here, imagine I'm doing a mistake. I'll do a purposeful mistake. Let us see how I can do mistake. So I'm just doing something and I'm just clicking here and it's going to add somewhere here. See, this is a mistake I did. Now for that, you have to select the region of your mistake. Okay. You have to select the region of your mistake. And once you do that, you can go to build and there is a option to delete. So I think sketching a molecule using a 2D and 3D using this build option and 2D using this edit 2D sketcher option is something that you can try upon. And additionally, the third thing is very important where you can do derivatives very quickly. So you have to click one atom, only one atom of your choice and go to build. Okay, build, the same build, add fragment. And in add fragment, there is something called as create enumerated entries. So this create enumerated entries is an option which will give you access to 2098 fragments. So here it is 2098 fragments, diverse R groups, 43 fragments. Whatever groups you want, you can click and activate. And once you have done it, click on enumerate molecule. Okay. So this is the third part. Okay. And once you have done this, now this is very, very important. Okay. So from now onwards, whatever job you will be launching, it should be in CPU. The reason is close to 90 odd students are here and we only have 8 processors. Okay. So I request every one of you to run the calculation in compute CPU node. Compute CPU node. So if you change that, then you can run this and click continue. So this will create around 2098 fragments from this single molecule for me. Okay. So one shot it is going to create, I think that much is sufficient for the 3 tasks. Give a try. I know I am raising the bar. Okay. Can RNA of 5, no, see, this is not a nucleic acid panels. Okay, so please let me just see. So the task now is 2D sketching, okay, and 3D sketching. Just give a try, and then small molecule enumeration. It's all options are there in build only. Okay. Yeah. Yes, Shikham, I'm like, you want me to like repeat which part? Did you found that panel or? Okay, so in 2D sketching, please keep in mind you have to do one attachment and do one fusing and do one deletion. Okay, similarly in 3D sketching, you have to make sure the options are in atom mode and then only you can attach and fuse. And then finally, you have to create a small molecule enumeration using compute CPU. Okay, that is not in localhost, compute CPU. So if you do that, then the calculation will run. Okay. Just give a try. See, give some time, play around with these three things. Imagine nobody's there and you have to sketch some molecule. And similarly for 3D sketching and creating an enumerations. So I'll be waiting for more questions. Yes, Shreya. So if you notice, the enumeration panel options, if you have to make sure you select one terminal atom, okay? So if you do that, just click this one atom and in the build, see the option is add fragment, create enumerated entries. And in that you have to choose a library of your choice and make sure you run the calculation in the job setting options. And please request you to change it to compute CPU 320. Is it okay, Shreya? Give a try and let me know if anyone of you faces any issues. Thank you, Jyotsna. Dr. Julie, thank you. See, again, here it is not a race. I request you, please focus on what are the core concepts I'm trying to convey. Okay. So we have focused on 2D sketching, 3D sketching, and doing enumeration. And associated options, all the options are available in front of you. It's our responsibility to just view it and make sense whether this option is going to be beneficial to me or not. Thank you, Niyati. Please, anybody else, if any of you have any troubles. Otherwise, I'll just quickly show you how to analyze this enumeration. So if you notice this enumeration panel output will come here. Now I have to explain the concept of selection and inclusion. So right now, see, this is in yellow bar, like, sorry, not yellow, it's blue, like highlighted, right? Can you see? And blue circle is also there. So blue circle is the place where you can include the entries. Inclusion, this is inclusion, and this is inclusion, okay? So if I do control click, this is multiple inclusion. And here, this is selection. So the entries that are selected are different from included. Okay. So you want to select this full group means enumeration. You can shrink it also like this. So this is the full group. And this group molecule, I don't want to see them one by one. I want to see all of them at once. So you have this option of window and 2D viewer. So this 2D viewer is a broad scale where you can, in the right side, change view columns. Okay, close to 10, make it, and then you will be able to visualize the number of molecules that is being enumerated. Okay, so give a try. Again, I'm repeating, make sure it is selected. Okay, all these molecules are selected. Understand the difference between selected and included. Okay, this is included and this is selected. And once you have understood that, do enumeration and open the 2D viewer panel. So 2D viewer panel will show you, okay, change the view by columns so that you get more structures. Just give a try for this also so that you can analyze how many derivatives you have created. Is it okay with everyone? Unable to select the structure. Why is it something like nothing? Selection is like this. Okay, so click here on the title. And if you click the whole group, it will select the whole group. Just give a try again, Swapnil. Thank you. So click on window and 2D viewer, then you can see all the structures. Okay, that's great. So I will quickly move on. Anybody want me to hold for some more time or shall I move on? Repeat enumeration. Okay. See, if you take this structure, Nupur, click on any terminal atom. Okay, any wherever you want to do derivatives and go to build. And in the build, we have this option of create enumerated entries. And if you do that, then you will see lots of fragments and derivatives, and you can choose what you want and make sure while launching the job, you change the settings to activate compute CPU and run the calculation. Okay. Is it okay, Nupur? Yeah. Anybody else? Okay, so I'm moving to the next topic, which is my biomolecules. Another biomolecule, we already see a DNA, and now we will dive into a protein moiety. Okay, that's interesting. So the PDB ID that we will be discussing is 6B2Q. So I think everybody knows the PDB has a 4-letter code. This is Aurora kinase dual inhibitor, Aurora kinase A and B for anti-TB. So 6B2Q is a target PDB ID, and if you click download, so you could notice that a PDB has been downloaded. That's nice. Okay. So whenever you see a protein of your interest, what is the first thing you have to do? you have to understand what is the composition of this. It consists of around 8,140 atoms. Okay. And then it has around 1,059 residues. And it has 4 chains. 2D sketcher, but under. Okay, it's fine. Don't worry, no issues. Okay. But if you understood, that's okay. That's fine. Yeah. So see, you will not get immediately everything. It's just I'm placing it. It's like a platter. Watch it later, whichever dish you all like it, you can eat it again. So don't worry. Okay. So shall I move forward or you want me to wait?
How to get the protein selected?
Yeah, no, I'm just checking here. I think Katya. Okay, yes, okay. Thank you. So once you have downloaded the protein, you have to investigate what is the composition of this PDB. So in the bottom, okay, in the bottom you can see like how many atoms are there, how many residues is there, and all that. And then the next thing is if you activate this window and sequence here, you remember we saw for DNA, right? Similarly, you can see here also. Okay, so how many chains you have? Chain A, B, C, D. But whether it is a heteromer or a tetramer, all sequence are same, right? See, same letters, all of them are. So that means it's a monomeric unit having 4 units. Okay, so we call it as tetramer. Now, if that is the case, mostly in drug discovery, until unless if it is not the interface of the protein two chains, so mostly we will be working on only on chain A. Okay. So in the left side, see, this is a new thing that you will be doing. In the left side, if I press protein, I can see chain A, chain B to wow to see domains in that protein. This is secondary structure information. You have domain view also separately in a multiple sequence viewer also. Okay. Is that a question or it is something you are happy about? See, Rakesh? Okay, so whenever you see a protein, okay, if you see in the left side, okay, here is a structure hierarchy. Till now, we have not explored this, right? How? How to see domains in the protein. Okay, okay, that's nice. Okay, so the how is nothing but, this is nothing but the sequence you remember in DNA. Yeah. Okay, in DNA, sequence viewer can be activated and deactivated like this. Same also. Is it okay? Saravanan Singh and Rakesh. Okay, so once you have done that, you will be able to see the ligands and you will be able to see the protein. Okay. Now, as I said, it's a multimer. I don't need other chains, so I will do right click and delete atoms. Okay. And then similarly, right click, chain C and delete atoms. So you are deleting single one by one chain. Okay. Now, similarly, for the ligands associated things also, you have to delete. Chain B, right click and delete atoms. Chain C, right click and delete atoms. Okay. So you should not delete anything associated to chain A. So this is chain A associated ligand, that is CJJ and OBD. Okay. Now I think the purpose of sequence viewer is done, so I will remove them. I have deactivated it and I'm able to see the company A with the 2 ligands. Okay. For proteins, I want you to activate the ribbon, which you did like same. Okay. For protein alone, don't do it for full. And for ligand alone, you have to activate the CPK form. This is the CPK form representation and OBD. OBD also you have to activate the CPK form. See, can you see there are 2 ligands? Okay. So this is ligand 1, which has an ID of CJJ and OBD. Okay. And you can also do protein surface also, like you did for your DNA. So do only protein surface. And the question to you is, all of you is, whether CJJ is the inhibitor OBD is the cofactor, or OBD is the inhibitor, and CJJ is the cofactor. Can you identify which among them is the inhibitor and the other one is cofactor? So this is the question I have for all of you. So please, so this is the task 6B2Q, okay, has to be downloaded and then delete other chains from structure hierarchy, okay, all except for delete all except for chain A, okay, and associated ligands. Okay, for downloading, I'm going to show it again in Office, except for chain A and associated ligand. Let me just type. And then you have to visualize ribbons, surface view of protein. And then you have to also see ligands, CPK representation. And then you have to identify whether CJJ or OBD is the inhibitor. Okay, so that is the question mark I have. And someone asked me to show the download. It's simple, click file and get PDB option. So you have to give 6B2Q and click download. Okay, so ask this question, okay, how do you identify? Now, just by visualization, what is your perception? Okay, so please tell me if you visualize this structure. So according to you, and see which is what. See, if you click here, you can see it is getting highlighted. If you click this, it is getting highlighted. Get a feel about it and tell me which is the inhibitor. So this time you have to give the answer with the CJJ, why and why not. Okay, please. Thank you. Rakesh, I did not got your question. How to again back to native structure? Native in the sense? What is native? Like normal, just you have to hide like this. You can deactivate and activate. Anybody else have any questions or answers? I've already placed the question in front of you. I'm seeking for your answer. So observe this. This is a hands-on workshop. So please observe and tell me if OBD is the inhibitor or CJJ is the inhibitor. Structure hierarchy is in the left side, in the structure hierarchy, okay? So here is the structure hierarchy. In case sometime like it may be like hidden like this, I will show you. It may be like where you can't see it also. So you can drag it up like this and you can see them. Is it okay? Yeah, thank you. Anybody else? Hello. Yes, I didn't got the answer. So according to you, by visual inspection, so what do you feel? Whether CJJ is the inhibitor or OBD is the inhibitor? Anybody having any answers, or shall I reveal the answer? Hello, am I audible? Or I'm having, yeah, okay. CJJ, thank you. Yes, may I know why? Yes, Tanya, that's a nice answer. It has a very good hydrophobic enclosure, CJJ, when you where this is solvent exposed region. Okay. And if you go to RCSB protein data bank also, you will notice the inhibitory profile of CJJ, not OBD. That is the answer. Okay. So we'll have a 4 minutes break and then by 12:30, we will resume if there are no questions. Anybody having issues, they can raise it now and I will be there to help you. Dear participants, don't hesitate. I think I have introduced you to the Mastro with a task-based learning concept, okay, which I have used, and I'm still learning many of the computational chemistry application. So anybody have any doubts, questions, or concerns, they can ask, raise it now. ligand CPK Johi, this is straightforward. So if you see here in the left side, okay, structure hierarchy has ligand tab. I think my screen is not visible. Let me just share it now. So if you see here, this is the screen, and you can see in front of ligands. Okay, so here is the CPK is the space filling model. You also have ball and stick, and you have tube form, and CPK form is the space filling model. Is it okay, Dr. Zuhi? Yeah. Anybody else have any other questions? So thank you all. I think there are no questions, so I hand over the mic to my colleague Kaushik. Kaushik, the stage is all yours.
Close the cloud instance.
Yeah, I have to close it. Yeah, thank you. Yes, I have logged in. Can you try now?
Hold on.
Yes, Soujanya, that's a nice question. That will be covered in the how this inhibitor and all that, how docking and everything in the later part. Yes, that also will be covered. Don't worry. These questions are very good. So Dushyant has asked how to know who is ligand and who is inhibitor. Yeah, Dushyant, you can go to that RCSB protein data bank, and usually in the associated literature, they will mention that the inhibitory profile of it. By visual inspection, this is a little guess that the buried ones are the good inhibitors. But Dushyant, that's a nice question. In RCSB, you will be able to see the inhibitory profile of it. Okay.
Yeah,
So
I'll address those questions first. Don't worry. Yeah, I'll take them one by one. I'll show them everything one by one.
yeah, thank you.
Thank you all for the questions. Keep the questions coming in. This is beneficial for everyone in the workshop. So please post your questions in chat box, and we'll address them one by one. So by now, I hope you're all comfortable with the Maestro interface. We have half an hour before lunch break. So I'm going to take like 5 minutes to recap what we had done so far. I hope all of you can see my Maestro interface. Let me just try to share again, just to be sure. Yeah, I hope all of you can see it. Can anyone confirm? Yeah, thank you. So what we had done, if you noticed since morning, we did a few things. So I'll just try to recap them one by one so that if anyone has missed those things, these are not really important for the rest of the things that we're going to do. We just want to get you used to the software. So the first thing that we did was we went to file and then we changed the directory to whatever directory that you wanted to create. Right? We named this day1_demo. You may have a different name for your directory. So it doesn't really matter what name you give as long as you know where you're keeping the files. So this thing will save all the files for your project in this directory. So you don't have to keep searching where the files are later on. The second thing that was done was we went to file, save project as, and we saved the project so that this is like similar to your Google Docs where everything is auto saved. So once I have saved the project, I added these molecules to my project. So if I close this, open the project, I'll be able to see all of these things again as it is. I don't have to keep saving them one by one. So let's say you have a power cut and you lose your data. You don't want to lose your data. So this thing, if you change your directory and save your project, then all the files for that project will be saved in that directory. And the project also will have all the things that you have done so far up to that point. Once these two were done, then he showed you the workspace, and this is the workspace navigator. And you have a section called structure hierarchy here. So for the workspace, it's this blank area where we're going to visualize the molecules in atomic level detail. The first thing he did was he went to the build option, and then we went to other edits, and we went to build biopolymer from sequence. We built a DNA molecule, and bDNA is a form of DNA. So DNA can be in a B form or A form. It can also be a Z form. We don't support Z DNA construction here, but we support B and A DNA. And you can also build a peptide or a RNA if you're interested in doing those things. But for now, we'll just focus on the DNA. Give a sequence here, and then you want to build a double stranded, so it's going to automatically complement the base that you have, A and T and C and G and all of that stuff. Once you click on build, you get the DNA that was shown in the workspace. Once you get the DNA, he showed you how to look at the surface view and the interaction toggle, and all of these things were shown to you. You're able to look at the hydrogen bond interactions, the pi-pi interactions between the different bases. So all of that was shown to you. And hopefully, by now, all of you also have an understanding as to how to rotate your molecule, how to zoom in and zoom out of the molecule. If you don't know how to do these things, there are different ways to do it. If you have a mouse with you, a mouse has a left click, right click, and a scroll wheel. If you roll the scroll wheel forward, it's going to zoom in. If you roll it backwards, it's going to zoom out. Similarly, if you want to rotate a molecule, you have to, the same scroll wheel, click on the button, click on the scroll wheel, hold it, and move the mouse around to rotate your molecule. And if you want to translate the molecule, you right click, and then you try to move the molecule anywhere you want it to be. And lastly, let's say that you have zoomed in the molecule too much or zoomed out too much. Okay, I can't really see where the molecule is. You can just click on this button with the 4 arrows, and that will always bring it to the center, no matter where the molecule is. If it is in this corner, you can just click on this in the center to bring it to the center, right? So these are some simple mouse actions. If you don't have a mouse with you, then you can try to use a keyboard shortcuts, where if you go to view option, you can see that you can press the K key on the keyboard for zooming in and the J key on the keyboard for zooming out. You have to press it continuously to see the zoom in and zoom out. If you want to rotate, you go to view again and go to rotate. You will see that you have this keyboard shortcuts like forward slash, semicolon, comma, full stop, square brackets to rotate in different directions. Same thing for translation. Also, you have other kind of keyboard shortcuts where you have these things. These tools are much more convenient to use if you have a mouse with you. So if you don't have one, try to procure one in the future. So once you have got control of how to rotate your molecule in the workspace, then he also showed you how to sketch a simple molecule. So you go to edit, and then you go to 2D Sketcher. And in the 2D Sketcher, you can try to sketch any group of your interest. So you have a few basic elements on the left. You have a periodic table here that you can load by clicking on this button. Then you can increase charge, decrease charge, single bond, stereo bond, double bond, aromatic bond. And you have a few cyclic rings here. So you can easily use any of these options to sketch a molecule. For example, in this case, he has sketched this molecule, and I'm just going to like quickly erase it. I can click on the eraser and drag a box around it. And I can show you how to sketch like some other molecule, like click on a benzene, click here, and then you want to add a bond, click on the bond here, and click on, you see that when you put my cursor on this particular atom, there's a circle that appears. Click on the circle, and that will draw the line automatically. And again, if I click on this circle, it's going to draw the line. Click here, it's going to draw the line. Click again, it's going to draw the same at the same atom 2 attachments, right? And you can keep extending this molecule as you want it. And so it's quite simple to do these things. If you want to change any of these groups with some other functional group, you can click on the element of your interest and click on that atom, and it's going to give you this OH. If you have a single bond, oxygen is supposed to have 2 bonds. But if you have a double bond, if I make this as a double bond, and if I click on the oxygen and click on this terminal atom, you'll see it's as a single O. It doesn't have the hydrogen because the valency is satisfied. So it's smart enough to realize if you have a double bond or a single bond, what is the valency supposed to be. In the same way, if I have a amine group, if I click on this nitrogen and click on this particular atom here, you'll see that it's going to add 2 hydrogens to make sure your valency is satisfied with the nitrogen atom. Then you click on Save as New and load the structure. So it's as simple as that. But I won't do it right now because I have this molecule already sketched in my screen. Then he showed you how you can do the same thing with the build option, where you can try to add fragments in the build. And if you choose any particular atom in this list, in this molecule that you want to replace, this particular option will appear, create enumerated entries. Once I click on this atom with the left click, you will see a blue circle around it. Now I can try to make derivatives of this molecule with this option. And he showed you how to make this large number of derivatives in this case. And all of these are derivatives of the same molecule with different substitutions. And if you want to look at these changes, you can just click on this circle and you'll notice that some changes were made, I think, in this region of the molecule. And so you can see all these changes one by one for these molecules, right? Yeah. And also notice that there's a triangle. Whenever you have this big group of molecules, I have around 1,843 molecules. So if I want to see all of them, I have to keep scrolling this thing up and down of these things. But let's say I want to work with the protein right now. The last molecule is a protein. I don't want to keep scrolling this one by one. I just want to like go to the protein directly. So for certain things where you have a group header, you can just click on this triangle in front of it to collapse a group. See, now I can directly see my protein PDB. Or I can expand the group if I want to look at the individual structures here. Then we loaded the protein molecule by going to File, Get PDB, and then we saw how to load that PDB ID. Once we got the structure, then he showed you how to look at the ribbon form by going to Style, Ribbon. We also seen how you can change the protein to a surface view by clicking on the surface option. And you can make the ligand in this CPK representation as well. So keep in mind that when you click on this brush option here, it shows you different ways in which you can show that particular molecule. For small molecules, I typically like to use the ones in the second row. Like you have the wire representation. When I put my cursor, it tells you what the representation type is: apply wire, apply thin tube, apply thick tube, apply ball and stick, and apply CPK. And if you want to change the colors, you can try to play with the colors here if you're interested. For proteins, I like to show them as ribbons or surface. That's what has been done in this case. So if you observe, just by looking at these simple things, we have covered the structure hierarchy. We have covered the workspace navigator. We have covered the workspace. We have covered the interaction toggles here. We also covered a few buttons in the file section. We have covered a few buttons in edit. We have covered style. We have covered build. We have covered this button also. So slowly we are getting used to all the different things in the interface. So now what we're going to do is we're going to move ahead for performing our docking calculation. So as I mentioned, for a docking, there are 4 things that you have to do. The first thing is you have to prepare your protein. And the second thing is you have to define the binding pocket of in the prepared protein. And then once you have done that, you have to prepare your ligands. And then finally, you dock your prepared ligands into the binding pocket of the prepared protein. Okay, so these are the 4 steps that you have to always do for any docking calculation. We're going to address them one by one. So the first thing is prepare your protein. So why do you have to prepare the protein? So if I look at this PDB, and this also pertains to the question as to how you determine if it's an inhibitor or a more substrate. So if I go to this PDB, right, and search for 6p2q, it's going to take me to this PDB website where I can look at the information pertaining to this particular molecule. So in this case, this protein is talking about dual inhibition of protein kinase A and B in Mycobacterium tuberculosis. So we're going to understand that there are a good bit of information on PDB. We're going to go through some of these aspects to understand what exactly does the structure contain. So when you're trying to work on your own targets, typically, you'll have to have a starting point. And if you have a protein structure as a starting point, that is well and good. If you don't have protein structure, there are other methods that you can use, but that can be complicated. For the workshop, we'll try to keep it simple. And we will try to take into account the structures in the PDB. So there are 6 things that you have to consider when you are trying to download the right PDB ID for your protein. The first thing is let's say, for a protein that you have searched for, if I search for, if I go to a PDB and I search for protein kinase A, and I click on that, and I'm going to get the hits for this thing. You can see there are 63,000 structures that are relevant for this kind of search, protein kinase A. So out of these 60,000 structures, which PDB should I use? Should I use like 1J91? Should I use 5J5T? Should I use 5WNL? It's too complicated to figure out like what to do in those scenarios. So there are 6 criteria that people often use to shortlist the right PDB ID for your given project. So the first criteria is organism. So do you want to target a protein from humans, or do you want to target a protein from a different species? Whichever species you want to target, make sure the organism indicates that. So in this case, we're trying to target this particular protein in Mycobacterium tuberculosis organism. If you want to work with a protein from a different organism, see if you can find a structure for the protein for that organism. The second criteria that you want to use is mutations. So in this case, there are no mutations for the protein. Ideally, you want to keep something that has no mutations, unless you're looking at, you want to study the effect of mutation on the ligand binding. So most of the time, you would want to work with a wild type protein without mutations, but sometimes you may want to work with a mutant protein, especially if you're keen on looking at the mutation effect on your binding. In some cases, if you don't have a non-mutant structure for the protein, then you can take a mutant structure and try to reverse mutate it to wild type protein structure, if you know what the sequence is supposed to be for the wild type. But for now, you can see that in this case, the organism is correct. The wild type, it's a wild type protein, it's not a mutant protein. And then the third thing you have to look into is the resolution of the structure. So in this case, the resolution is around 2.88 angstrom, and this structure was determined by X-ray diffraction. The other commonly used approaches are NMR spectroscopy and cryo-electron microscopy to determine protein structure. In this case, this was done with X-ray diffraction. The resolution of 2.8 angstrom indicates how good your structure is. The lower this number is, the better your structure is. So 1 angstrom resolution is better than 2 angstrom resolution, which is better than 3 angstrom resolution, it's better than 4, and so on and so forth. So let's say after doing the search, you have identified the organism, you identified a structure with no mutations, and you still have left with multiple structures after that. You take the one which has the best resolution. Okay. So resolution is the third criteria that you have to look into to identify the right PDB ID. The lower the number, the better the structure. The fourth criteria is you can look at the structure in the PDB itself. So we go to structure tab here. And once the structure loads, then you'll see that there are two different colors available for you. There's a pink colored one and green colored one. And if you want to find out the difference in the colors here, if I just put my cursor on this pink one, you can see there's a small label that appears on the bottom. So if I look at this, it says serine threonine protein kinase pKNA. Just below that, it says 6p2q, model 1, instance ASM_1. Then it says D. D indicates chain D. And if I put my cursor on the green one, it shows you that it's a chain A, right? A, it says ASM_1, A, which indicates the chain A. So how do I know which chain to pick? Do I have to take chain D? Do I have to take chain A? So the same protein, same structure, both have a ligand bound to them. And I don't know whether I have to take chain D or whether I have to take chain A. So if you look at the structure, you will see that some parts of the protein has its dashed lines. Right? There's 1, 2, and 3 dashed lines here on the pink chain, and you have 1 and 2 dashed lines on the green chain. So these dashed lines indicate missing regions in your protein structure. Even if the structure is deposited in the PDB, there could always be some areas where the protein structure is not known exactly. So those areas are shown as its dashed lines, but they don't know exactly where the atoms are supposed to be. And so they just like leave it as blank. Because chain A has fewer dashed lines, which means it has fewer missing components, I'm going to take chain A to go forward with my work. So take the structure with fewer missing components. The pink one has 3 missing components, 1, 2, and 3. The green one has only 1 and 2 missing components. That's why it's much more reliable to start with this structure. And if you go back to structure summary again, you'll notice that, these are the 4 criteria: organism, mutations, resolution, and number of missing components. The less the better. For resolution, I just want to point out a couple of things. If you have a resolution of more than 0.8 or 1 angstrom, if it's higher than that, most likely your structure will have missing hydrogen atoms. So if you don't have hydrogen atoms in your structure, then you don't have the full chemistry of your protein. So it won't be, you have to add those hydrogen atoms before you can go with your docking calculations. So just looking at resolution, I know from experience that if it's more than 0.8 or 1 angstrom resolution, then that means it will not have hydrogen atoms in the protein structure, and you'll have to add hydrogen atoms to it. And if you go down further, so for every structure in the PDB, you always have a paper published with it. You can see that in this case, they're talking about this MTB, which stands for mycobacterial tuberculosis, PKNA, PKNB, dual inhibition. So this tells me that they're trying to work on inhibiting this protein, and any molecule that is bound here should be an inhibitor. And if I go down further, and before I do that, so this tells me that typically I have inhibitor bond structure here. So you can have two scenarios. One is, as I told you, every protein has a substrate that is the naturally binding molecule. And you have the inhibitor that is trying to inhibit the protein. So if you have a multiple PDB IDs which has a ligand bond to it, you have to take the one which has the inhibitor bond structure and not the substrate bond structure. Okay, that is the fifth criterion. So in this case, since I know that the paper is talking about inhibitor design, so most likely it will be inhibitor bond structure. And I can read the paper to confirm if it's inhibitor or not. So typically for the inhibition, they'll talk about Ki values, which indicates that it's a inhibitory constant. Ki stands for inhibition. For substrate, you probably have a Ka or Kd values. So that's a different thing altogether. So if you have Kd, things like that, it could be a substrate, it could be inhibitor, you don't know. But if it says Ki, it's most likely inhibitor constant. And if you go down further, you'll see that it also lists the two molecules that are bound to this protein. You have a molecule named OBD, which the structure, you can see the formula here, you can see the structure here. And you have a molecule named CJJ, you can see the formula here, you can see the structure here. So how do I know which one is the main molecule? Some of you said that the depth of burial of a molecule indicates that is the main molecule of our interest. That is perfectly good way to analyze it. A second way to find out is that if you look at the PDB itself, it tells us bottom binding affinity annotations. And the binding affinity is reported only for this CJJ molecule and not for the OBD molecule. So this tells us that CJJ is the main molecule of our interest, and it has a binding affinity of 9 nanomolar in this complex. So if you have like multiple inhibitor bond structure, take the one that has the inhibitor that is close to your chemistry of interest. So these are 6 criteria that you want to use when you're trying to shortlist a PDB ID from the protein. So if you don't have binding affinity values reported, from experience also know that typically these carbohydrate looking molecules are not good inhibitors. So if you see something like this, which is like very polar, typically this won't be a good inhibitor. That's from experience. That's why I know that CJJ is better, but I also know it because I have this affinity value reported for CJJ molecule and not for OBD. So in this particular PDB ID, we have a high resolution, which means that we'll have to add hydrogen atoms to the structure. We also have 2 missing loops that we have to fix for the structure. And we have this the second molecule bound to it. So this is the third issue that we have to fix. So first is because of 2.88 angstrom resolution, it won't have hydrogen atoms. So we have to add them before we go for docking. The second issue is that it has 2 missing components in the protein structure. So we have to add those as well. And the third issue is that we have 2 molecules bound to it. And we have to remove OBD molecule from this complex because it is not the main molecule of our interest. So for these 3 issues to be fixed, we'll go back to the software. And since he has already loaded the molecule, I know I'll just ignore it for now. I'll try to load it from scratch. So let's say I go to File, I'll go to Get PDB, and I'll change the 632Q PDB ID here. And I'm going to give the chain name as capital A because I know that I don't want to take chain D because D has more missing components. I'm going to just choose chain A here specifically, File Get PDB, chain A, and click on Download, and that will load your protein with only the chain A. It's much more simpler way of doing this than to load the whole protein and delete the other chains from there. So try doing this, go to File, Get PDB, and download only the chain A for this protein, 632Q PDB ID, and chain name has to be capital letters, capital A, and click on Download. Once you have done that, please ping the chat box saying done. So please run the PDB ID and click on chain A, enter chain A, click on download. Once you have this, just ping in the chat box saying done. I'll wait for 1 or 2 minutes. Once you have done that, we'll go and run our first calculation, which is the protein preparation workflow. Yeah. I can see some of you are able to do it. I'll just wait for 1 more minute and then I'll show you the task to perform. All right, let us continue. So once you have got the chain A, I can see that if I go to this protein here and click on the style and click on ribbon, you'll see the protein in ribbon form. And for the ligand, I'll just click on this brush for next to ligand, click on apply CPK, and that's going to show the 2 molecules for me. And if you're not sure which one is what, if you put your cursor on each of these molecules, you will see on the bottom, it tells you what that molecule is. So if I put the cursor here, you can see it says OBD302, which is what we have to remove. And if I put my cursor here, it says CJJ301. This I have to keep. I have to remove this. And second thing is, if I also look at this protein structure, if I zoom into it a bit, you don't have to do this, I'm just showing you on the screen. You will notice that there are different colors for the atoms, right? So you have some blue colored atoms, you have some red colored atoms, you have some gray colored atoms. The blue colored atoms are typically nitrogen atoms. So you can see that when I put my cursor here on the blue colored ones, it says N on the bottom. And the red ones are oxygen atoms. So red is oxygen, blue is nitrogen. This is the default coloring scheme for most of the software. So keep in mind that if you have a dark blue color, it is nitrogen most likely. And if you have a dark red color, that is oxygen most likely. Carbons can be different colors. In this case, the carbon is in gray color that is being shown here. And if you look at this blue colored atoms, you can see that nitrogen, this particular nitrogen has only 2 bonds present for it. It's supposed to have a 3rd bond, which is not visible right now because it doesn't have hydrogen atoms in this structure. So once we add the hydrogen atoms, it's going to have an extra white line that indicates the hydrogen atom for this structure. Okay. So the whole protein has these issues. It's like most of this protein doesn't have hydrogen atoms present in it. So we'll have to add the hydrogen atoms for this. And we also have to add the missing loops. And we have to get rid of the second molecule that we're not interested in, which is the OBD molecule. So to do this, we will go to this panel called Protein Preparation Workflow. You can see where my cursor is. Just click on this option called Protein Preparation. And you will see a panel that looks like this. If you can't find the panel, just see my screen again, it will be located here. Protein Preparation is the button. Just click on that button, it's going to open a panel for you that looks like this. Once you have done this, then we will address these 3 issues: missing loops, missing hydrogen atoms, and getting rid of the second molecule. So first, we will remove the second molecule. Then we will add hydrogen atoms. And in this case, I won't add the missing loops because it will take some time and they're not really important for this protein. And I'll tell you why later. Okay. So to remove the second molecule, I will go to this, in the preparation workflow, you have 3 different tabs: preparation workflow, diagnostics, and substructures. I'll go to substructure tab, and you'll see a button called load workspace entry on the top left inside substructures. So click on load workspace entry. It's going to analyze all the components of your complex. You have the protein, you have the 2 ligands, CJJ, and OBD. When you click on this row, it's going to highlight each of those molecules for you. And sometimes you can also have water molecules as part of your PDB structure. If they are present, they're going to display it here. In this PDB ID, there are no water molecules, so we don't worry about them. But sometimes you may have to deal with water also later. So right now, I have gone to substructure tab, click on load from workspace. Then I select this molecule, this row, which says OBD302, and I'll click on delete from entry on the bottom right. Once I do this, now I only have 1 protein and 1 ligand. I don't have the other ligand present. And you can also see that in the table also it shows only CJJ and protein. And on the left, it gives me a new structure named 6p2q with deletions. I will repeat again. Once you open the panel, go to Substructure tab, click on Load from Workspace button, select the row with OBD, and click on Delete from Entry. Once you have done this, come to the first tab, Preparation Workflow. Any questions so far on this before I discuss the preparation workflow? We'll take 5 more minutes and then we'll break for lunch. We will launch our first calculation. We'll go for lunch after that. Okay, if there are no questions, then if you go back to preparation workflow, you will notice that you have a few settings to go through in this section. The first setting is, if you go to global settings here, you have the option to define your pH for the environment. It is set to 7.4 right now because it is the biological pH. The pH of blood is 7.4. If you work in a different environment, different pH, make sure you change the pH here. And that's going to affect your protein structure in the charges on the protein structure here and there. But now we're going to use pH of 7.4. And you'll also notice that just below that says small molecular process. In this case, I have a protein structure, along with it, I have a ligand bound to it. It's going to identify the ligand automatically. If you have metal present in the structure, it's going to identify metals and ions also. And all of these are required, are going to be called as HETs, H-E-T-S. Yeah, so you don't have to worry about anything here. So you just mention your pH, change the pH, and the rest of the options keep as default if you're interested. In this case, we'll keep the default pH and the default options here also, nothing to change. Once you have done these things, then the next section is you have a pre-process section where you have option to fill in missing side chains. So this will fill the side chain provided you have the backbone present. And if you go to more options, you'll see that you have a few more options here, like assign bond order. So a molecule can have single bonds, double bonds, so on and so forth. So if you want to figure out the right bond order for these molecules, just check this box, which is on by default. So a good rule to follow is that anything that is checked on by default, keep them on always. And the other ones that are not selected by default, you can add them if you're interested, or you don't have to add them if you're not interested. So these ones that are enabled by default have to be kept on always. So if you click on assign, if you put your cursor on assign bond orders, it shows that it's going to assign missing bond orders based on geometry or chemical component dictionary, or really assign all bond orders using CCD. So when you put your cursor on these things, it gives you a slightly more explanation, a longer explanation in the yellow box next to it. So if you click on replace hydrogens here, you can see that it shows remove any original hydrogen atoms before adding explicit hydrogen atoms to the entire structure. So if hydrogen atoms are present, it's going to remove them and add its own hydrogen atoms. If they are not present, it's going to add hydrogen atoms automatically. So this will take care of our second issue, which is missing hydrogen atoms for the protein structure. So we have removed the second molecule. Now we have taken care of this issue of not having hydrogen atoms also by this option here. If you have metal present, it's going to create a coordinate covalent bond between the metal and your coordinating groups here. If you don't have metal, it's going to skip this option automatically. So don't worry about having this on or off. If you have two sulfurs close to each other, it's going to form a disulfide bond. You can look at other options also in this case. If you want to fill the missing loops, you have to enable this option. It's going to take some time. So to avoid that, I'm not going to do this thing because in this case, I know that the missing components are far away from the binding pocket. So I won't worry about it. But for your own projects, later on, when you're trying to work on your own projects, make sure you enable this option of filling missing loops and check if the loop hasn't filled or not once the structure, once you get the output from protein preparation. So I haven't changed any options in this whole more options section. Just keep it as it is, nothing to change here. Then once you added the relevant components of side chains, hydrogen atoms, all these things, you may have to optimize the position of these atoms. So you have a section for optimize H-bond assignments. Then eventually it's going to minimize the entire structure and give you a prepared protein structure. So nothing to change in these sections, just keep the default options. So all I have done in this that panel is I removed the extra molecule and I kept the default options. Now I'm going to run the first calculation. I'll give a job name here so that if I come back 1 year later, I know what the job was for. I'm going to prepare 6p2q and I'm going to prepare the chain A of 6p2q, not the entire protein. And then before you click on the run button, keep this in mind for the entire workshop that anytime you click on the run button, before that, click on the wheel next to it It's going to open a panel that looks like this. Change the local host to compute CPU 320 and click on Run button here. If you have done this correctly, just click on Ignore if you get any warning like this. If you have done this correctly, you will see that it says Job Started here. And then on the top right, you have a Job button where you can click on this. It's going to say the job is waiting. It will say waiting, then it will say running, then it will say completed. Okay. So if you have done this correctly, then everything will be good for you. So I'll stop my screen share. All of you try to launch the calculation and then break for lunch. So you have to do this. Once you load the protein, open protein preparation workflow panel. Go to Substructures tab. Click on Load from Workspace option. Choose row with OBD molecule. And on the bottom right, click on Delete from Entry. Then go to Preparation Workflow tab. Look at the options, don't change anything. Give a job name. Go to Job Settings by clicking on the wheel next to the Run button. Change localhost 8 to compute CPU 320. You have to do this for the whole workshop anytime you see a Run button. Then click on Run in the Job Settings panel itself. So there are a couple of questions. One is, can you do protein peptide docking also? Yes, you can do peptide docking as well. That's a different way to do it. We'll discuss that later if time permits. If the ligand is a ligand deletion, we can do it from left side option directly. Yes, you can do it from left side also. Do not have to use the panel for that. Both are equivalent. If the protein structure is not present, then only sequence is present. You can use a tool called homology modeling, which is not going to be covered in the workshop. You can use this tool to predict protein structure based on sequence. So launch the calculation, and if you have done it, you can break for lunch. We'll come back at 2 o'clock sharp. If you don't see the job launched, then please let me know. I can fix it for you right now. Yes, you can do RNA docking with protein. That's a different panel altogether. We'll discuss those things maybe at the end of tomorrow's workshop, if time permits. But right now, we are focusing only on small molecule docking with protein. But in principle, you can dock a protein to a protein or a protein to RNA or a DNA. But those are different methods altogether, and we have to explore them in a separate event altogether. So for these other questions, drop an email to me. You have my email ID, and we can try to discuss this other queries through a separate Zoom call for your particular project. Yeah. We can model RNA molecules as well. We have published some papers on that front, but typically we would prefer if you have a starting structure present from some repository, because RNA structures are quite complicated to model, and they can have very diverse structures also compared to proteins. I hope all of you have launched the calculation. If you haven't done it, if you need help with it, please let me know. If you have done it, break for lunch. We will end the Zoom meeting shortly. And we'll come back at 2 o'clock sharp, and we'll continue with the rest of the demo sessions, where we're going to look at defining binding pocket. And then we'll prepare ligands. Then we'll go for docking and analysis of docking results. So please do this. If you need help, I'll wait for another 5 minutes. And then I'll conclude the Zoom call. We'll come back at 2 o'clock again with the same Zoom meeting. Any questions? Just click on the run. Once the job launches, you can break for lunch. I hope all of you have saved your project. If you haven't done it, please do save it by going to File, Save Project As. If you see the name on the top saying like docking.prj or some other name.prj, that means your project is saved. Don't worry about it. If you don't see this _.prj on the top, and if you see Scratch project, that means your project is not saved. If it's not saved, go to File and Save Project As. If you see a name here on the top, that means it's safe. Don't worry about it. Yeah, Dushyant, those are the complicated questions with RNA molecules, right? So I don't have a good answer for those things. If you have something like 10 bases, we can try to model it appropriately. If you have something more than that, it becomes complicated to get a good structure. But there are other resources, online tools that are available for RNA structure building. You can try to use those as a starting point, and then once you have a structure, then you can try to import it into the panel, and you can work through that. So if you have a protein structure that is not in the PDB, you can try to either build a structure from any software that is available online or within the software of Schrodinger. Once you build it, you can load it into the software itself by going to File, Import Structures. I'm going to explain that later in the post-run session. Any other questions? If there are no questions, then I'll close the meeting. Please come back by 2 o'clock sharp and continue with the rest of the sessions after that. Thank you.